Welcome everyone to the Sound Toxins training and meeting for 2025. We're so glad so many of you were able to make it in person, and for those online, welcome as well. Hopefully you'll be able to enjoy this experience as well. My name's Michelle Lepori-Bowie. I am the program manager. And this year we also are welcoming Melissa Petrich, who is our new
I'm a water quality specialist.
Water quality specialist at Washington Sea Grant, and also helping out with Sound Toxins, co-managing the program. Hi. Okay, and this is Melissa over here.
Oh, yeah.
For those on.
Yeah, hello.
Okay.
Great. So first, we just wanted to cover the Sea Grant Code of Conduct. So I don't know if people are familiar, but we just want to make sure we're treating each other with respect in this space. So the main parts of it are that we treat everyone with respect and dignity. We share perspectives and offer feedback openly and honestly. We're open to experiencing new concepts and differing viewpoints. And we work to ensure that others have opportunities to speak and share. Also want to take responsibility for your actions and impacts, acknowledge harm when it has occurred, and apologize. We want to also report perceived unacceptable behaviors directed at yourself or others to Sea Grant, so that's to us. But if you want to see the full code of conduct, you can scan the code, but that's just kind of how we want to treat each other in this space. And so if we can all agree to treating each other that way today, that would be amazing.
Thank you.
Great. So we did want to do a quick introduction of sites and of everybody here. So if we could just go around the room and say your name and your pronouns, the site that you work at, and the years of experience you have with phytoplankton. We'll do everyone in the room, and then we can move to folks online. My name's Melissa Petrich. My pronouns are she, her. And I don't have a site, and I did some phytoplankton work in undergrad, but otherwise I haven't done a ton. So I'm a big zero, but I'm excited to learn today.
Let's go this way around the room, and folks online, I hope you can hear, but sorry if you can't. We don't have a microphone to pass around, but just go for it.
Site down in Illinois, and I first started with phytoplankton with Anthony and ORHAB in 2018, and then I've been with SoundToxins for about 2 years. I'm Kate Hubbard. I am visiting from Florida. Okay. And so I don't have a site, but I've been working on phytoplankton since 1980. The Nancy Stephens, Los Angeles. We're starting our 3rd year.
And I'm Tamara Galvin, also from Karenia Sparrow Marine Life Center, and we've been SoundToxins for about 3 years.
I'm Amelia Calder. I'm the 3rd person in there doing Port Angeles. I'm new to this, so about a year.
Get that table next.
I'm just taking over for someone else, so I have 0 experience.
Welcome.
My name is Mary Kelly. My site is Quartermaster Harbor on Island, and I was living in this, I think, since 2012.
Awesome.
I'm doing and our sites, we have 3 sites in Central Tucson, Liberty Bay, Dyson, and
We'll go to the back table next.
My name is Christopher.
Yeah, that's a goal.
I'm Real Foster with the Beach Product. I sample at the beaches, Laylock beaches and Blue Beach, and I've been to the Anthony Odell, University of Washington, ORHAB, I'm the post coordinator program out there, because I've been doing this since 2000.
Should we pull up the chat? Or let people do it. Okay. Hi, folks online. If you want to unmute yourself and introduce yourself, and say your name, sites that you monitor, and the years of experience you have with phytoplankton, you can do that now.
This is Jennifer Sand. Can you hear me?
Yes.
Okay, I'm on Whidbey and I stare at the Camino Island people thing on Zoom. And I learned about diatoms way back in the dark ages, mid 70s, when I was at Froggy Harbor Laboratories on San Juan Island. And since then, I got to be a 2022 Sound Water Steward and just kind of slipstreaming on the Camino group.
Wonderful. Thanks. Anyone else online? Yeah, it's not mandatory, but you can also share in the chat if you prefer.
Good morning. This is Rebecca Cohen. Can you hear me?
We can. Hi, Rebecca.
Hi, good morning. I just wanted to say hi. I'm with Taylor Shellfish. I know we've had staff that's participated in some of the sound toxins efforts before, and so I'm excited to be in on this training this year. So
Welcome. And we also have Chris in the chat, who is from Kitsap County for 5 or 6 years with the group over there with 3 sites. Thanks. Welcome, Chris. And yeah, anyone else?
I'll say hi. Hi, everybody. I'm Tracy Berry with the biotoxin program at the Department of Health. Hopefully seeing you in person tomorrow. And I've been working with phytoplankton for 10 years, but mostly I work with the toxins and the results that you send our way.
Well, hope to see you tomorrow, Tracy. Okay. Sweet. All right, awesome. Thank you, everyone. So that kind of gives us an idea that we have a lot of different levels in the room of experience, and that's wonderful and okay. And especially welcome to the new folks who are just starting. We love to have new people. And so hopefully you are all able to either attend or watch the recording of the introduction to phytoplankton we did 2 weeks ago. But just in case, I will do a very, very quick review, so it hopefully will be fresh in your mind. But also first, I suppose we'll go over the schedule for today, which is that we are just going to be spending the whole day looking at HABs under the microscope. So we'll have Kate and Anthony. We also have several cultures. So we'll do a little bit of PowerPoint presentation to talk about things, and then we'll go around with the cultures and do a little drop on everyone's slide and give you a little bit of time to look at them and see if you can see what you were supposed to see on what we tell you you're supposed to see through the PowerPoint presentation, if you can see that in person on the microscope. So that will be the schedule. And we have not officially decided like what order, so it'll be a fun little surprise. And we plan to have like a little coffee break. I know I apologize that we seem to be out of coffee already, but we will be trying to rectify that before the coffee break. And then we'll have lunch at 12. So I know this is going to be like a little bit awkward to have all of our microscopes and like toxic species and also be having lunch in this room. So what we'll try to do is like scoot everything, I don't know, to the middle of the table and eat carefully. And we also have access to the outdoor space if it's not raining. It might be a bit wet, but at least you can go out and get a little fresh air. So yes, that's just the situation, the logistics. And we'll take a short break in the afternoon too, just so that we don't all go crazy, but hopefully it'll be really fun for everyone. Any questions?
So we have one person.
Uh-huh. No, you should, if she has the link, she should be able to jump right on. And if there's an issue, we can, I don't know, email her the link or something. Really? I thought I saw her earlier. But yeah, she should be allowed. Okay. I guess let me know if she can't get back on in a couple minutes, but we'll move on for now. Oh, and then just for those of you that are going to be here tomorrow, we have a series of talks. Oh, this isn't our updated. There's a slightly updated version on your tables that you can look at with just a couple of changes, but overall it's going to be mostly different presentations about research projects and updates, and hopefully some time for questions and answers as well. And also if you have materials to exchange, if you come in the morning, hopefully we'll be able to give you some stuff and take your samples. And if you are not staying and you need your stuff today, just see me or Melissa separately and we'll get you all set up. Okay, so a quick review of some taxonomy terms, especially for the new folks. So we'll be looking at least one diatom today, and then several dinoflagellates. I think there's only one diatom, if I'm remembering correctly. But just as a reminder, diatoms are our phytoplankton that have glass frustules. That's the case that encases them. And what matters when you're looking at diatoms is which direction you're looking at them at. So that if you're looking at them from the like top or bottom of the petri dish in your head, the top is called the valve. And so if you're looking at it from the top or the bottom, it's a valve view. And then if you're looking at it from the side, you have the two thecas or outside valves of the two valves of the diatom are connected by a lot of girdle bands. So we call the side view the girdle view. The top of the cells are called epi and the bottoms are called hypo. So for diatoms, dinoflagellates, we have this orientation that we look at them at, where the top is the epi and the bottom is the hypo. So for diatoms, the epitheca is the like larger part on the top, and the smaller one that fits inside is the hypotheca. For centric diatoms, generally they'll have symmetry, radial symmetry, and often they're circles, but they'll sometimes be ovals or various other shapes. Again, if you're looking at them from the valve view, you might be able to see that symmetry, which is the bottom circle here. The top one is the girdle view, so you might see like more of a rectangular type shape or a different type of shape, but just knowing which angle you're looking at them at can help you figure out what you're looking at. And then pennate diatoms are generally more elongated and have symmetry on their, what's this called? Sorry, my brain's not working super well this morning. Symmetry over a line, a wraith, or kind of like the central line that they go to. Anyways, if you're looking at them from the valve, they might look kind of like a boat, or there's various different shapes. They also have a girdle view that if you see them, they also, again, might look a little rectangular. It depends on the cell. And then rarely you'll see them from the narrow girdle view. That would be like if you dropped a bunch of pencils and one of them landed straight up on your microscope slide. That's what you'd be seeing. So it might look quite different, but you might see them at like a diagonal or an angle. So just remembering that these are three-dimensional shapes that we're looking at under the microscope. And then some other features to look for diatoms, for things that are in chains like Heterosigma, kind of where the connection points are. The space in between could be called the aperture. Spines or setae are things that are sticking out where they connect, and the angles are also helpful. And I didn't add, I was going to add a couple more pictures to this little review, but for some of the other diatoms that you look at, there's the features that stick out of them. They often have a lot of processes, is what we call them in English, or just little things, features that stick out of them, and also can sometimes connect them. So like a Thalassiocyra chain might be connected by these like long processes that connect them in between, but they also might have ornamentation on the valves that can help you identify them. So we will be spending a lot of time looking at dinoflagellates today. So just as a quick review, we have some unarmored dinoflagellates and some armored dinoflagellates. That depends on kind of how solid their cell walls are. So unarmored dinoflagellates are don't have like solid plates that give them structure. So they're just like a little squishier, and also can, which means they can kind of change shape a little bit. So there's a lot more variability when you're looking at them. But for most dinoflagellates, the way we kind of orient them to understand their features is that they have a ventral view, which I like, which means stomach. And I like to think of it, so it's the front of the cell, and you can also think that's where the two flagella come out of the front. So I kind of think of it as like a belly button, where their flagella come out of. So that's the front. And then the back is called the dorsal view. You of course might see them from the side as well. They're often going to be spinning or moving around. So just kind of looking at the front is where you're going to see most of the features that you want. Or if you're looking at it from the back, just knowing that everything will be flipped. Again, the top of the cell is called the epicone, the bottom is the hypocone. And then the band in the middle of the cell is also is labeled as a girdle on here, but we also call that space the cingulum, which is where the flagella that goes around the cell is kind of protected by the indentation. And then the flagella that is the kind of trailing one is also protected by a groove called the sulcus. And so if you're seeing the sulcus, that's the front of the cell or the ventral view. The other important feature for unarmored dinoflagellates that we'll talk a little bit about today too is that for all cells, you can see their external structures, but you can also, they're see-through, so you can see their internal structures. So you'll be seeing a lot of different organelles in these cells, including chloroplasts. Sometimes you can see the nucleus, and sometimes you can see what's called a pustule. And for some of these unarmored dinoflagellates, kind of the location of the nucleus might help you identify them as well. So knowing how to identify that can help you identify the cells that you're looking at. And then just a quick on armored flagellates will have kind of structures that we call plates. Sometimes you can see them. You can especially see them if you have like an empty or dead one. It's a bit easier to see, but even on some of your live ones, you'll be able to see kind of like more angles or more kind of like solid structure. They also have, we call it epitheca and hypotheca. They also have a ventral view and a dorsal view. The ventral view has the sulcus and where the two flagella come out of. And the cingulum and the sulcus are what we're often looking for to identify these. So speaking of the cingulum and the sulcus, I cut up this picture to kind of talk about a few different features. So where the cingulum is on the cell, if you look at the top left of the screen, if your cingulum is right in the middle, that's called median. And if it's above or below the kind of like halfway point of the cell, that's one way to describe, you can call it pre-medium or post-median, but that helps you figure out what cell you're looking at. You also, the sulcus generally goes down on this type of cell, but on occasion it can have what we call a sulcal intrusion, which means the groove extends up into the epitheca or epicone. And so in top right, if you're looking at the one labeled A, that's a little bit of sulcal intrusion, and on D, that's quite a lot of sulcal intrusion. And for some unarmored dinoflagellates, the sulcal intrusion can actually go all the way up to the apex, which is the top of the cell, and create a specific shape or groove that can help us identify it as well if you're looking at the apex or top of the cell. We can also look at displacement. So the bottom left here, basically when the cingulum wraps around the cell, if it meets exactly in the middle, for example, I should have put this one here. So can you see my, oh yeah, cursor. Okay, great. So here you can see that the cingulum kind of meets back where it starts. And so it's kind of a straight line. So that's no displacement. However, if it meets slightly differently below or above, it is called displacement. And we usually measure displacement by the width of the cingulum. So if it's, I kind of tried to guess here. So this is the width. This might be closer to 1. It's a bit hard to tell, but basically you take the width here and then how far down it is. I kind of felt like it was about half the width of the cingulum. Whereas here, if you measure this width and then go down twice, that's where this one reconnects. Whereas this one here is kind of a really big displacement. So it's probably like almost 3 or even more displacement. So that's how we measure that. Okay, and then we also have what we call overhang, where the sulcus is almost like covered because the cell kind of overhangs the sulcus. And then torsion is what it's called when you've got a cingulum that kind of wraps all the way around more than once and goes all the way around. So some things to look for. And that's all I have. Well, I guess I'll put up my thank you slide for now, but we'll now pass it over to, I think we're going to start with Kate this morning to start a presentation. And then when the cells arrive, we can start looking at cells. So Kate, would you like to come up? Oh, also, I guess any questions before we pass it off? Okay. Um,
I brought 2 kinds of material. I brought bluebell stick cells and I have live cells over here. And I have a few different species, and so we can kind of play with those and see. I don't know if you want to hand them out now, or maybe I can talk a little bit and then pause on the slide.
yeah, why don't you start talking a little bit and then we can pass the stuff out so that people aren't too distracted. Okay, so I will share the slide. And then should be able to advance it just by clicking here or here if you would like.
And then
Oh, geez. Yep.
I'm not
One
good
more
with
thing.
these things, so.
Thanks for catching that. No, I was gonna do that and
So,
there we go.
again, I'm Kate, I'm from Florida, but I went to graduate school at the University of Washington. And so I worked on Pseudo-nitzschia. And before there were sound toxins, there's different types of sampling, but I did a lot of my sampling using a wetsuit and going around and trying to figure out routes where I could take ferries and basically cover the whole sound in a day. So I have a ton of respect for all of you and what you do, and it's great to be back. Unfortunately, usually when I show up, it's not a good sign because I work on toxic algae, and that usually means there's something going on. And so for today, I'll give a little bit of an overview of Karenia species, and I'll talk about some of the different features we use to discriminate them. And I'll be using material that Celia Valak has presented at the National HAB training course, and she's adapted that material with Karen Steidinger. And then we also teach an internal course, and then I've peppered it with a few other things. And I've tried to include material that has a lot of detail, but I also will talk about just sort of general impressions. So hopefully, please stop if you have any questions. So I'll talk just a little bit more about the thecate dinoflagellates, some of the morphology that's important for Karenia, an overview of some of the species that occur, a little bit about the life cycle and asexual division, and that ties into some cryptic morphology. And then tomorrow I'll try to talk about some HAB detection and what we might do even with the samples from last year. Okay, so not all dinoflagellates have the same cell covering. We just heard from Michelle that there are thecate dinoflagellates, and then there are also armored or unarmored. And Karenia is really interesting. It is an unarmored dinoflagellate, and so that makes it special for a lot of reasons, and it can make it kind of problematic as well, because they are fairly squishy. And I feel like this is, if you look on the lower right, a really good example showing the variability in terms of the material within the thecal vesicles that ranges from the athecate at the top to the one that has a thick theca. So in terms of like Alexandrium, something like that, and they're going to form thick resting stages, and that's important. We don't necessarily have the same observations for the unarmored. And it's interesting to point out these microtubules here. I have another slide on those, but that's part of what allows these species to be so variable in their morphology. So all dinoflagellates are thecate, and again, you can just see some of the variability from an empty theca all the way to a sericium cell that's got a really dense theca with even plate overlap. So again, it's just an important distinction. And we already heard about this, but we have these unarmored and armored, and again, with the unarmored, we see a huge amount of morphological variability for all different types of reasons. And so that's just something that there's not as much of a constriction on their shape. And the other thing that we look at really carefully, there's a few different things that we look at for these dinoflagellates. Michelle pointed out a lot of them. And one of the things that we look at as a diagnostic feature for some of these Karenia species is the sulcus intrusion, and that's right at the top of the cell, and they can have these different patterns. I think I'll come back to that with another slide. These, this is an image just showing the nucleus and then those microtubules that I mentioned. And these are these tubular polymers and they form the cytoskeleton and they allow a lot of that morphological variability. So they give the cell shape, but also allow it to move. So these are really fascinating and they're energetically, it takes a lot of energy to make these. And so it's, again, it's interesting. In terms of the ultrastructure, the nucleus, as you can see, it's this dense material in these cells. It varies quite a bit. And with a lot of the athecate, or the unarmored dinoflagellates, we do tend to look at the nuclear placement. And it is condensed and has several hundred chromosomes, so it does usually have a pretty obvious appearance. And it's one of the main features that we use to look at differences across the Karenia species. So it's the nuclear placement within the cell. So these are some examples of the apical grooves and the epitheca. And again, these are really important for differentiating Karenia from a lot of the other genera that look like Karenia. And it's got a nice straight groove. That's something that if you get a cell just in the right image, you can sometimes see that in light microscopy. And we have 10 known Karenia species and at least Karenia brevis, which is really famous in Florida and the Gulf of Mexico. It has really only been identified in this part of the Atlantic, but we have a lot of other Karenia species that do exhibit global distribution. And we also know that at least sometimes our Karenia brevis blooms do get transported out of the Gulf of Mexico, but we don't think there's another source. We have started to, whenever there is a weird Karenia bloom, we usually get involved somehow. And so we have seen some other blooms. For example, we got samples from New Jersey from a few years ago. They saw discolored water and they had cells that looked just like Karenia brevis. And it turned out we did a bunch of testing. It was Karenia papilloniacea, which is another toxic species, but it produces orders of magnitude less toxins than Karenia. But in terms of what's been observed, you'll see that there's not a lot that's been observed on the West Coast. There have been blooms of Karenia mikimotoi observed in Alaska. So it's kind of an interesting conundrum as to whether or not Karenia may be here, but it's not terribly surprising just given the distribution all over the world. There are a number of different species and only a few of them are known to produce brevetoxins, which are the really nasty toxins that can become aerosolized. They cause neurotoxic shellfish poisoning, but there are some other toxins identified for some of these other species. And so in general, for looking at Karenia with those 10 described species, there's the straight apical groove, there's a nucleus, and we'll look at that again in another slide. And there's some different components here, but these aren't really going to help you so much looking under the light microscope. This is the ventral view, and so that's something to be mindful of when you're looking at these cells. You can see the cingulum band is displaced, and this is Karenia brevis, and you can see a nice straight sulcus here. We generally can divide Karenia species into these two shapes. One is the Karenia brevis shape and one is the Karenia mikimotoi shape. They tend to have slightly different shapes. So the aspect ratio may be able to help, but keeping in mind that they have variable morphology, the main difference is their carinas up here. So Karenia brevis has this curved one and Karenia mikimotoi has a straight one. The nuclear placement is also really important. And so here is a great table, and this has all of the different species, and it also shows their nuclear placement. And so this is really critical because, for example, if you look at brevis versus brevisculata versus longicaunalis, you can see that the nucleus is in different parts of the cell. And again, that's one of the main features that we can use to discriminate these species. Also, if you look at these, you can see that some of the cells are longer, some of the cells are broader, some of them are almost spherical. And so that's another feature. But I would say that with blue galls fixative, iodine-based fixative, you'll be able to see these different nuclei. And I think that would probably be the easiest way to discriminate. This table is also great because it does include toxins. This isn't a book chapter, but it mentions some of the different toxins. And again, only a few have been observed to produce brevetoxins, but there are some other compounds that are produced during Mikimoto blooms, some of the other blooms that are not well described, but they have shown to be have negative impacts on benthic organisms, for example, and there can be really huge blooms. Some of the blooms that occur of Karenia Mikimoto can be millions of cells per liter, and that alone can make their morphology change too. So I have some details about each of the different species. I'm going to go through these pretty quickly so that we can actually look at the cells. But if you're interested, I'm happy to go through any of these. But Karenia brevis, again, tends to be more of the rounder cell. The nucleus is round and it's in the left hypothetical lobe. And so that's something that we really look for Karenia brevis. And it has numerous oil bodies and chloroplasts. And, you know, it looks hugely different depending on the day, the time, what's going on with the bloom. And let's see if I can get this to play. Here's a little video just showing the cells swimming. They do have their two flagella, so they're able to kind of move around in that tumbling motion. And that's something that's very characteristic. I think that they're really cute when they're doing this. So it's always helpful to see them moving around. And for us, we have these blooms that start every year. They start offshore, they get moved onshore by currents. We suspect that there's both aggregation caused by the currents, but also cell growth. And it's really hard to know kind of what's going on with the bloom, but it's possible that there could be sort of a similar situation here. The brevetoxins can be aerosolized or concentrated in shellfish. Generally, finfish doesn't accumulate toxin in the muscle, so if it's cleaned well, it's okay to eat, but we do advise not to go harvest in an active bloom. Our blooms can cover hundreds of square miles. They can endure for over a year, and they cause huge negative impacts on marine mammals, sea turtles, humans. There's a lot of respiratory irritation that gets reported. And so we have a huge network to monitor these blooms. And since 2019, we've also reinvigorated a state task force. And as part of that, we've gotten funding for Center for Red Tide Research, and that just helps fund a lot of the different monitoring that we do. Okay, so that's all for brevis. And then Karenia Mikimoto, this is probably one of the most globally distributed species and the most commonly distributed species. And it has bloomed in Alaska. It started to bloom in New England. And we do see it sometimes in Florida. And this one tends to be, you can see there's a little bit more singular displacement here. And the cell tends to be a little bit more oblong. The nucleus itself is also oblong, and you can see that it's occupying the left side of the cell. So it's generally much bigger than the Karenia brevis nucleus. So those are some of the features that we use to really discriminate these two. I'll come back. This one is very similar to another Gymnodinium species, and I have a few examples of Karenia-like species at the end. So Karenia papilloniacea. This is one that is almost, it has a lot of morphological variability, and it does look a lot like brevis. And there's been sort of a continuum of brevis to papilloniacea shapes observed. And so it can be hard to definitively identify this, but it's got a slightly pointier epitheca, a slightly broader hypotheca, and then you can see the nucleus is in the same place as in brevis. So that's not really a great feature to help differentiate from brevis. Longicanalis is a little bit longer of a cell, so we can see that pretty clearly. And then you can see the nucleus is right in the middle. Celliformis, this is another one. It's got a little bit more of a defined shape morphology to it, less singular displacement, a more defined hypotheca, and then you can see the nucleus here is sort of spread across the hypotheca. And then here's another species, Asterochroma. This one's a little bit more condensed. It can be a little bit longer. And you can see the nucleus is actually spherical and in the epitheca. Okay, so here are some of the different ones that we see in the Gulf of Mexico. And these are all fixed with Lugol's so that you can see that nucleus really well. And again, these are the ones that are toxic with respect to brevetoxins. We don't know about this one yet in terms of the genetics. We have started to do a lot more genetic work with these, and we found that some species are collapsing into one. So some of that sort of normal taxonomy with phytoplankton. One of the things that we don't know about, so I know that Alexandrium makes these resting cysts that reside in the sediment. And in terms of the Karenia brevis life cycle, we have observed sexual reproduction, but we still don't really know if there's a resting stage at the end of a bloom. And for Karenia mikimotoi in China, there has been some work that suggests that there may be a resting stage. But we're not sure if that resting stage is a long-term resting stage or a short-term resting stage. And it's pretty rare to find it in the sediment. And it's something where we've started to use a lot of genetic tools to look in the sediment to try to see if we can identify this missing life cycle stage. And we've also been using the imaging flow cytobot to look at Karenia cells, and this allows us to process cells, use AI to help diagnose which species are present. And we have been able to identify 3 different morphologies of Karenia that we're tracking for brevis. One is our typical cells. Even with the typical cells, there's a fair amount of variability. Dividing cells, so these are cells that are actively dividing, and then these round cells. And these round cells we kind of equate with potential stress, but we don't really know, and we think there's a lot of different reasons that they can go round. So sometimes if you take those samples and you put it on ice, they will go round. Sometimes if you shake the sample too much, they will go round. We found that for IFCB, we had a copper intake hose, and they didn't like the copper, and so they were going round. We also find that at the end of a bloom, we had a big bad bloom in Tampa Bay several years ago, and we saw the whole population turn to these round cells at the end of that bloom. And so in terms of trying to look for something in the field, it's a lot easier to see those vegetative cells. The round ones, who knows? But when we've been tracking a bloom for a long time and then see everything change, we can identify that. And just as a note, these round cells, you can see a few of them do still have their flagella. So one big question is whether or not these round cells can go back into their typical vegetative morphology, and whether they can swim, divide, do all the same things that the vegetative cells do. For all intents and purposes, they do likely still contain brevetoxin, so there's still a problem in terms of toxicity. Whether or not these reside in the sediment, we don't know, but it's something that we're trying to look for. And I think that's important when you think about the source of cells and what might be the source of cells if you have new events. Because it could be from an existing population, or it could be transported in via currents. So yeah, so we'll see if they liked the trip here. I did bring them in my luggage so that they had like minimal disruption. And I brought the fixed cells just so we had something to look at for sure. Even putting a cover slip, like I can kind of, if you're just putting the cover slip on, you can squish the cells. So be pretty careful when you put the cover slip on these guys. Okay, and then really quickly, Karenia brevis and Karenia papilloniacea can have overlapping morphology, but they are different species. Karenia longicaunalis, Karenia umbella was recognized as a separate species, but it's just been put in as a synonym of Karenia longicaunalis. So those are actually the same species. And then Karenia digitata was just moved to Carlidinium. So, you know, as we get the genetics sorted out, there may be some additional surprises. And there's another genus, Brachydinium, and we see this kind of variability in the morphotypes. Where offshore, we see more of these Brachydiniums. This is a sister genus to Karenia. I think they're really, really neat looking. But at the top, you can kind of see as we go through this morphological change, and we end up with, you know, this weird elongated Brachydinium phase. In reality, it was suspected that these are all the same genus, but they're in fact quite different. Okay, this is what's going on in Florida right now. So this is an example of our Karenia data, our monitoring data and dots superimposed on a remote sensing image. Karenia brevis is usually visible from space once it gets going in around 100,000 cells or more. We are using the imaging flow cytobot on routine surveys to help map out these blooms. So these maps are coming to us each day while they're out on the cruise, which is new in the last year, and it's huge. So we don't have to wait for people to be out for a week, come back, and then look at all the samples on the microscope. We still do that, but at least we know what's going on. And this is what you might expect to see from an aerial image in those areas where we have kind of the red dots. And so this bloom is very, very obvious. But again, this is once we get over 1 million cells per liter. And this is our current bloom. But when I say it starts offshore, we've been tracking this since we had our 2 big hurricanes pass through at the end of September and in October. And if you just follow kind of the yellow, you see that this bloom has moved around a lot. It's been kind of shifting to the south. And unfortunately, it's still around. We still have these intense bloom patches. And so we're kind of concerned. We don't know if this is going to go away or if it's going to keep going. A few other unarmored dinos that are just good to be aware of and could show up. And I'm just going to go through these really quickly. Takayama. And again, it has this nice curve here. Carlidinium, there are 15 described species. These can be morphologically similar to Karenia. Gymnodinium aureolum, this is one that can be very similar to Karenia mikimotoi. And Karenia mikimotoi had not been observed in New England prior to 2017, but this species had. And so there's some confusion as to whether or not there's a mix-up in the literature. But this one can look very similar, but still has some differentiating features. And then this is another gymnodinium, but again, the shape can be kind of similar. So I thought it was helpful to just show some of these, and that's it. So I was gonna put this, maybe this one up. So this has, I have several of these species. I have brevis, papilionaceae, mikimotoi, and longicaunalis. And so we can hand those around. And again, some of the main things to look for are the nucleus, trying to think about which orientation of the cell you have. You can try to squash them and see what they look like, you know, squashed, non-squashed, and we have live and fixed cells. They probably will not swim unless you have a slide that has a reservoir. So, and any questions? Let's look at some cells. So even when you pipette these guys, we'll want to pipette them kind of slowly. They're not sensitive. If you're looking at the large one like that.
So are you gonna be able to share your slide, your,
Yeah,
okay.
sure. Well, I mean, let's.
Or should I leave this up for?
Yeah, I mean, so I have several.
So for folks in the room, there are a couple of extra microscopes. So if you don't have one, you can share with folks around you. There's also, I have an extra one that's not being used. We'll also, Anthony, on every table, there is flat slides. But Anthony has a couple of extra deeper slides, so we can distribute those around if you want to look at the sewing pattern a little bit more. But be very careful with those, as you all know, those are a little bit more expensive, so be careful. And then folks online, we're gonna leave the slide up for now, but we're gonna try to hook up a microscope camera in a little bit, so just hold tight. Great.
Okay, take that one too.
Do I need to go around and pipette, or should just giving you five bars? Okay.
Yeah, but not much. Yeah.
For anyone who's like brand new, if you want like help with pipetting or anything, or just a little extra help, just like raise your hand and wave it around and someone will come to you.
And just a quick note, I passed out the Karenia brevis and Karenia papillonacea, so all the similar morphologies to start out with. And then I have the other ones in my bag.
Stealing people, pick over people's shoulders. What's on my?
On the left. Thank you for having us. You're really fun.
So you're at the US one?
In 2000.
So I was in but I was, I've been a sea grant since. I'm not. It's entirely possible. And then no, it's what you got.
It's
Yeah, now we're talking about the leakage and compression salt. Yeah. It's interesting here, you have two different agencies, it sounds like the field. Interesting. Yeah, no, it's been very moderate. Very expensive. Yeah. Yeah.
So I think I just have to stop sharing and then you can share. You should have permission to share. Actually, share the screen with the folks online, but let me see if I can find a resource for you. Okay, well, 1 second. Okay, try now.
Well, that reminds me of that.
This is where like different types of fixatives affect other cells differently, which is why like formally my top guys other things that it's really well informative that might change their shape. And Amber Garland, so I never do another warranty, but like this time I drive by.
Take a picture of that from your lecture. I don't want to mess you up on the camera, but
Are you looking at about the
yeah,
Papillonicia? Papillonicia. Okay.
yeah, let's take a peek. Hang on just.
Yeah, for sure.
So. Pretty sure. Second, you read, seeing that the character is 2019, having a central coast and Two days before I received a photo from somebody else. A week later. And then also. It's like a lot of.
Unfortunately not.
I do see that in our records, especially for people that they felt, you know, the way they get
So if you could try to kind of come up there for a month and I came back and everything. So like back to.
Yeah, we don't know if it's like a very short period that they go into and. It's still very much. But the idea is. We don't. We see the regular budget. We see it all the time. So it's always present in both organizations. So there's always a source of budget and stuff. Okay, sorry. Be a friendly source. Maybe we just have a friend. Yeah, I mean, well, they probably are able to interact with the subject. Other people have reported that they swim down in the sediment and come back up if you wait to get new.
So we had a question online and I wanted to address it in the room as well. The reason we're looking at these today is because we have actually seen them on the Pacific Coast. The ORHAB program reported seeing a couple at several sites last summer. And anything that we're seeing on the Pacific Coast can wash into the Sound at any moment. So though we currently don't have these commonly in our water, there is a chance they could be there, and that's why we're training on it today.
This is
I should have started with that, sorry.
No, I mean, I probably should have said, someone online said, so is it bad news that you're here? And I said, kind of. It's concerning. We don't have a bloom, huge bloom, but we have.
You're being precautious.
Yeah, we're being precautious.
And these Mickey motoi look to me like little tiny Akashiwo. How do they compare in size if we were to see them both on the slide?
Okay, that's a good question. Akashiwo compared to
Mickey
I think they're bigger, right?
bigger.
More spread out, and they generally have like that beautiful. But
like twice the size of the Mickey motoi?
I think there can be variability in the in their sizes too. I've seen a fair amount of.
Yeah.
I think we have Akashiwo, so maybe we'll do that one next, and then we can compare them.
They look very similar to me in their body shape and the way they move too.
Yeah. The other thing you might look for is, I think Akashiwo doesn't have a groove, straight groove that goes up to the sulcal intrusion. I think Akashiwo doesn't have that.
Okay,
So that may be
that
a thing
for
to look
sure.
for.
Okay.
And I'd have to look up what Akashiwo's nucleus looks like, but that might be indicative too.
Kind of shaped
It's dorsal ventrally compressed, but it's like shaped like this. As well.
Okay. Great. Can you say that again to the whole room in a bit? Also, I think we have an Akashiwo culture, so I thought that would be a good one to move right into after this, but make sure everyone. No, it's, we have a small amount in a cooler that we'll probably like go and pass around to folks. We have a lot more, so many cultures, thank you. I mean, so much volume.
I was just trying to understand about like duplicates of every table, so they had some of them in cooler type and some of them.
That's amazing. I didn't actually look in the cooler yet, but I think it's gonna be like one flask that we're gonna have to go around and pass out rather than handing out to everyone. It's a little bit different style, but this is great. Yeah. Did we announce that the coffee's refreshed?
No, but there was some stuff.
Okay. Oh.
off, maybe it's like the pressure or something. Oh, okay. But it's also like, I don't know, it's probably better coffee. We could say there's more coffee.
There is more coffee for those in the room if you are waiting on it. There is more coffee.
All the parking is full. Just park car, not that far, but like, yeah, like, yeah. I was like, oh no. It was so surprising, everyone's coming in from far away. Yeah, and yeah. There's lots of things that I stay out. Yeah. This is so good. I know. I don't want to take some pictures. Then I got distracted and I didn't. Oh.
Anthony, what are you looking at right now? On the screen is Karenia Mikimotoi, and earlier, what was earlier? The first one that you saw on the slide was Papilloniaceae. And currently, it's Mikimotoi. Yeah, I'll type it in the chat too. Elvis. That's tricky. Yeah. Anthony, if I move my computer near you, so people online can hear you talking, and Gary, why don't I mute myself and then you can unmute yourself? Okay. Okay, we're just gonna mute then.
Just for reference, there are 5 species and we have both fixed and live material. So if you want to see something that you haven't seen, just wander around and grab it off another table.
Okay, hi, folks online. Sorry, that was a really long, we had a lot of fun here looking at these Karenia species, but we're getting ready to move on to the next one. So, here is Anthony, to talk about the next species. I'm gonna stop sharing on your computer.
Yeah.
For you.
Okay, hang on. We're going to stop sharing on my microscope computer and switch over to the laptop here. This one. Okay. And then share this one.
Share screen. Point, share. Here we go.
Okay. Hi, Anthony Odell, University of Washington, ORHAB. I think I have done a few of these online. I don't know if you guys remember, when was the last? July, I think we went over all of our local HAB species over 3 days. Just basically that talk. But what we're going to look at next is Akashiwo sanguinea. And we're kind of moving, from Karenia into this because both of these are called gymnodinoids. So they're kind of these naked flagellates. So you can see it has a great degree of morphological variability. That's all Akashiwo sanguinea. And again, gymnodinoid, this type of cell that used to be all called gymnodinia, gymnodinium, has now been broken up into Akashiwo, Carlidinium, Karenia, and how you tell the different genuses apart is not just based on morphology, but you also look for the apical groove complex, which is kind of this sulcal intrusion into the epicone, and then it can be like straight or kind of squirrely. In this case, has like kind of a little curlicue. Yeah, apical groove complex that would be a great name for a band. It's one way to remember it anyway. So these are much larger than the Karenia species that we're seeing. Apparently the culture is quite healthy. And I think we're going to see kind of the morphology up there on the top left, my picture. These cells are the ventral dorsally compressed, so they are flat, so they do kind of like to lay on their back generally. They're also somewhat sigmoidal in the same degree, and they have this large kind of rib that goes from the apex down to the cingulum and the epicone. And if you adjust your focal plane up and down, you can kind of see that rib. And this kind of where are we? We're right here. And so it's not always visible, but it seems like in our area, it's quite common. Again, this is it's a very large cell. Yeah, the hypotheca is bilobed. You can see there's a bit of go back. You can see that there is a bit of a sulcal, go back, intrusion into the hypocone. And so it kind of makes the bottom this, it's either rounded or can go to points. Akashiwo sanguinea is responsible for fish kills. It produces like a kind of a soapy substance that can affect the gill tissue. It's a very fragile cell. And so if there's any kind of wind or surf disturbance, it'll break up. And you'll see like an orange bubbly foam, looks very soapy. It will turn the water quite orange. It's a visible HAB species. In 2009, we had a really large bird kill on the outer coast. Common meres, cormorants, there were several birds that were affected, and dead birds like littered the beach. And I was up doing some event response and sampling, and I went out to La Push, and nobody was surfing, but it would just look like this perfect glassy waves, and I was going to go try it out, and I could see this out there. And at that point, usually when it comes to HAB species up here anyway, it has to be like concentrated in some kind of vector species, and just going out in the water, it's not going to make you sick. I went out and I ducked my first wave and, was breathing out my nose. So it was fine. Paddle back out, taking off on the first wave, and everything was like really nice and smooth. And I'm like, oh, it's so soapy and the board handles so well. And I got in the barrel and I was going really fast and I wiped out. And when I went underwater, I opened up my mouth and I had like kind of water shoot in my mouth and out my nose. And when I came out the other side, it felt like someone had poured bleach in my nostrils. I was like, wow, this is like really, really intense. And first I was just like, well, maybe it's just like the speed and the friction. As I sat out there, my eyes started to water and I'm like, okay, I need to get out of the water. I went on the beach and the other surfers like, wow, like, did that hurt your ears and your nose? And I'm like, yeah, it did. So as far as I'm aware, I'm like the first kind of, known human, affected, directly infected by Akashiwo sanguinea. I did lose my sense of smell for about a month and never really came all the way back. Same with my taste. After that, in 2009, we had a US HAB symposium meeting in Ocean Shores. And so we had all of the experts from around the United States were there, including the CDC. And so we kind of put together a snap kind of meeting and we did some event response. We went over to Westport, which is a really common surf area. The balloon was still going. We did have low numbers, but we were doing nose swabs and ear swabs and talking to surfers, doing interviews before they went out. And then when they came back from the water, everybody experienced some small symptoms, put together a study. And so there are human health effects with Akashiwo sanguinea. So yeah, fish killer, bird killer, and can mess you up if you're swimming in it. So we have some great samples and we're going to put those around and probably going to take us a minute to get everybody
Yeah,
a slide.
we only have two, so we're just gonna go around and drop drops on your slide. So if you have your slide ready, if you want the well one, we can fill it, but if you just use the flat slide, we can just do a quick drop to look at. And yeah.
Be careful when you put your cover slip on because they will pop. Like I said, they're really fragile. I do have some Sedgwick rafter slides, the big well slides up here, and some Palmer Maloney slides. So
quite a few people had Palmer Maloney.
okay.
Yeah, maybe Palmer Maloney will be better for this one.
Yeah, because you can also see the swimming pattern as well, but really large cell, easy to identify. So and common. Yeah, easier.
Okay, yeah, I'll go do that.
Area. Yeah. Is it? Yeah, I just want to like look at your stuff. You have like all of the stuff in there. It's like full screen. Yeah, what's the next? So they have a raised field. They can use they don't do this. They say, yes, if it's moving like this, it's
But there's so distinct in its
It was meant to go. And then I ended up on my right part, but my problems were more.
Well, they get excited about it.
minutes doing it's like. That's where we get the most time about that.
I mean, that would be confusing, but also they wouldn't all be equally overlapping. So you have different areas to overlap. It's really rare to get completely stretched out. Yeah, usually like the colony will have a different part to overlap. You see what looks like it's starting to line in terms of the stack. What you're looking at.
No, I think we take photos and then we.
I would have just been like, oh, this is my fault.
It looks like a box. Oh, sorry. So the sample came from culture. It's doing really well. You can see the ventral dorsal compression. It's flat when it's swimming around on its side. It is somewhat sigmoidal, but it's not especially pronounced in this culture. Also, it doesn't really have that kind of mid rib and the epicone that's very pronounced. On some of these, you can kind of see it, but it's not very pronounced. So these size cells are just like they're not the biggest it could be, but they're all doing very well, and so they kind of have like the bodies are kind of rounded as well. And also, they has a bit of a in the epicone above the cingulum. It has a little bit of a flare. Again, Akashiwo sanguinea, one of my least favorite taxonomic terms, is morphologically variable. It has a really thin skin, and so it can change around depending on health and life stage. Usually when I see individual cells, and also Amy, I saw one in her sample that you had right here. That's kind of you can see that midrib in the epicone. So yeah, we do have another sample of it over here as well, and I hope to look at her sample this afternoon because it's just full of all kinds of great stuff. So yeah, everybody seeing it okay? Okay.
We have about half an hour before lunch, so we thought we might squeeze in a little Alexandrium. If you guys are all ready to move on. Sorry. Feedback. Okay, great. So in case I didn't announce this before, you can put like use flat slides. You can put like Kim wipes, pipettes, and cover slips into the container labeled waste, the bigger one. But any of the flat slides, you can like wipe them with a Kim wipe and reuse them, but also you can just plop them into the cup that says use slides and then get a fresh one. For obviously for your weld slides, you want to clean them out really well. But for everything else, you can just grab a new one and get ready, and we'll pass out the Alexandrium in a minute. And also Anthony will do his talk real quick, and then we'll pass out the samples.
Yeah, I think
That's in the first one, right?
in this,
No, I think
it's in the first talk. Yeah, it's in day one.
You can stop sharing your screen. Okay. You want to start on the slide?
Yeah.
Okay. So we will share slide. Here we go. Oops, sorry. Here we go. All right. Yeah. Okay.
Is this called all? Okay. Yeah. All right.
That is the first slide, because right before that. Okay.
Okay. So Alexandrium cantonella, I think if you from around here, you know that it has been here forever, right? So some of the first red tide issues that we had where people got sick that we know of back in the late 30s and early 1940s was up along the strait with Alexandrium cantonella. And also the very first time white folks showed up, George Vancouver was up off of Vancouver Island, and he sent 4 of the sailors in to get some shellfish off the rocks. And while they're in there, you know, they were pick one, eat one kind of thing. All 4 fell ill, and 1 died on the way back to the boat. So, you know, welcome to North America, Vancouver Island. And yeah, so it's been around forever. And the natives have known that as well. When these bloom, it's generally good kind of water conditions for other dinoflagellates, and those other dinoflagellates will exhibit phosphorescence, and they bioluminesce green. And when they see the green in the water, they would know not to harvest, which, you know, there's a good parallel. Water's growing green. Sometimes we get sick when that happens, so let's not do it. It's kind of the same conditions, right? It used to be just kind of like don't harvest in summer. Now we have Alexandrium blooms all around the state, all throughout Puget Sound, all on the outer coast, all year long. And so just a few years ago, was it 2010, we had a really large bloom on the outer coast. Now, something with Alexandrium was when conditions aren't right, they form a cyst and they drop down into the sediment where they can stay for decades and just kind of wait to get kicked back up and have conditions be right again. So once you have Alexandrium in your area, you have caught Alexandrium. If the tides and winds don't blow it out and it dies there, now you just have Alexandrium. And we saw this big kind of movement from kind of the strait into Puget Sound in the 1970s. Saw a really great toxicity back then, and then it increased in the 80s as well, and now it's spread to every corner of Puget Sound. In the 1970s, there was a big population boom in Puget Sound, and also there was a lot of septic and sewer. There's probably some anthropogenic nutrient loading that was going on. I'm just speculating, but they got around the corner. There was nutrients. They were happy. They spread. They died. They formed cyst beds. Now they're all throughout Puget Sound. There are several species of Alexandrium in our area, and I'm going to go through those. With taxonomy, as we've mentioned before, we usually just go based on morphology. What is it shaped like? And then based on that shape, you know, does it make you sick or not, right? Well, now with metabarcoding and all this genetic work that's going on, we're realizing that a lot of these things are closer related genetically than we thought. And so the names have kind of shifted and moved around. Some of those species, became varieties, and then they just disappeared altogether, and they kind of lumped. One thing with Alexandrium cantonella, which is we have here, is there's some morphological variability as well, depending on if you see it in single cells or it's forming chains. So hopefully in this sample that we're going to look at, we don't have just chains. Maybe we can see some single cells. Generally, and how you would tell the different species apart is you really need SEM, ideally. And you're going to look at the plate that sits right above the sulcus. It's called the one plate. And then you're going to look at the apex, and there's called the apical pore complex. And it's a little bit of holes and commas. And sometimes there'll be a pore or a little intrusion on the sulcal plate or the first apical plate right above the sulcus. You can, if you have time, you can squish these cells, but first take like some bleach water, like 50-50 bleach water, drop a little bit on the next to the cover slip of your slide, take a Kim wipe on the other side of the slide, and it will pull that through. It'll bleach all the organic material out. And so you'll actually be able to see the plates, and then you can take like a pipette or pencil and just press down on the cover slip, and it will break those plates apart. And I have used that to identify the species before. It is kind of a tedious trick, and you have to break a bunch of cells, but it's also kind of fun. So if you have time. So, I mean, this seems really complex. We're going to go based off of morphology. So this is kind of the big one right here. This is Alexandrium cantonella. It forms chains, and that's what helps us identify it. We have some athecate or lightly thecated dinoflagellates here that also form chains that also produce PSP, but this is heavily thecate, which means it has a very thick shell. They kind of look like little orange cheeseburgers. They're kind of squished anteriorly, posteriorly, so top to bottom. They have an excavated cingulum, which means it's cut out, and so that transverse flagella that goes around the body is like well protected. You can generally see on the posterior end, there will be just a little slight bump where the trailing flagella comes out of the sulcus, especially in individual cells. You can kind of see it in the line drawing up there. Yeah, this is the one that's kind of been around forever, and it has world distribution. It's everywhere. But now, again, with that world distribution, we're realizing there's some issues are arising based on morphology. So this is the big one. This is what our sample is full of. I'm going to quickly go over a few other species as well. Oh, that brown color. It can be like a rusty orange, red, yellow, brown. It also kind of depends on the filter you're using on your microscope, or if you're not using a filter on your microscope. There's lots of little round brown things, lurbits, little round brown things, okay? If you see something that's round and brown, does not mean it's Alexandrium. There's lots of things that appear like Alexandrium. So don't just automatically go there. This is one thing when you're doing taxonomy and you start with HABs, people see things that resemble it and think everything's a HABs. There's hundreds of thousands of species of diatoms. There's, thousands of species of dinoflagellates as well, especially even in our area. And so not everything that's little round brown thing is a Alexandrium. Okay, so Alexandrium acanthella, we know it was based on morphology. It seemed to always bloom concurrently with Alexandrium cantonella. Like you never saw one without the other. It's a variety, but then it turns out that genetically, they really are identical. It's a morphological variety. Here you see that the cingulum, that kind of mid-belt area, is post-median slightly. So the epicone is like a little bit rounder, they're a little bit taller. This is still Alexandrium cantonella. So you can see these in single cells, and they look like another species type, tamarense as well. So again, morphology makes this a little bit difficult. But for the most part, if you look at the cingulum, you have what offset of 1. And if you were to squish these cells and look at the one plate and the apical pore complex, you'd see that it looks almost identical to catenella as well. So I know this is not helping, right? Because it makes it more confusing. But this is just what we're up against here. So there's another type. There's another group type, tamarins. Tamarins tends to be a little rounder. The epitheca is going to be a little more conical, and it can have like a little like a protrusion, like a little hat, a little bump on the apex, which kind of makes it easier to tell apart. Again, the cingulum is highly excavated. If you saw this and you called it catenella, you know, for our purposes, that's fine. It's still toxic. There are some non-toxic strains, but then again, as we've gotten into metabaric coding and we've looked closer, it might not even be related to non-toxic ones, even though they're morphologically the same. So, yes. Yeah. So I mean that again, there's more morphological variability. So just Alexandrium SP, that's fine. You know, if you see one, you should be worried. So it doesn't, you don't have to see it in chains, you know, it can be some of these other species. If you see one, it's a problem. Alexandrium SP is fine, or Alexandrium CF means confera for you know, appears like, for reference, catenella. So Alexandrium CF catenella, that's also fine. Don't get too stressed about it. If you want confirmation on the species, you can send me a picture and I'll do my best. Send me 3 photos at different focal lengths if you can do like something that looks right on the surface, ventral view, like you've seen the textbook, that's ideal, obviously. But if it's laying the other way and you're looking at the dorsal side, if you can focus all the way through the cell to try to pick up the cingulum and sulcus where they meet, that can help me as well. Yes. Yeah. Yeah, no, that's totally fine. Morphotype, variety, var, that's all acceptable. And the more work that's been done on Alexandrium, we're realizing that it's really complex. And so it's just Alexandrium SP is fine for now. There's still a lot of work going on. Just going to leave that slide up so you guys can all read that yourselves. I mean, that I like this. This is an algae-based taxonomic note. Shrug, right? So, you know, and this is, you know, pretty up to date. We just know that it produces paralytic shellfish toxins, saxitoxins, and you only need a feel. And so if you see something that looks like this, that's enough. Yeah, I just got back from a course in Naples, the advanced phytoplankton taxonomy course. It was 3.5 weeks. And we spent a whole day on Alexandrium, and at the end of the day, we're all like, okay, thanks. You know, it's not even clear now, but also it was kind of to make the point that it's very confusing, a lot of work's going on, so don't get too overwhelmed. Austenfeldii, this one's much smaller than the other Alexandrium species. It is its own species, and you can identify it pretty well. If you look at the line drawing up there at the top left, it is generally a little rounder, so it doesn't have that compression either way. The cingulum is not very excavated. It's actually pretty close to the surface, and so a lot of times you can actually see the flagella around the outside, kind of going back and forth if you adjust your focal plane. When you preserve them, their flagella generally fall off. And there's also another point that I want to make is you need to look at live samples as soon as you can after you taken them. Looking at preserved samples alone, even if it's Lugol's or formalin, it's going to alter the cell. And you really want to see stuff live, and you can see greater detail, but you can also see it kind of swimming around, swing patterns, things like that will help you identify species. Non-chain forming bioluminescent. So again, if you see that green or kind of phosphorescing in the water. This could be one species that does that. Often blooms concurrently with Cantonella and other dinoflagellates that also phosphoresce as well. Mildly toxic. It's the least toxic of the genus, but still toxic. So even if you see one of these, again, just Alexandrium, SP, that's fine. Like these are a little bit smaller and rounder, so a little bit easier to tell from the other ones. Yep, okay, that's it. I don't want to get into the other float around brown things. But yeah, we'll go back to this photo, and then if we can hand out some of our culture, we can all get a look at it, and then I'll put some online and project it as well. Are there any questions? Lots of questions, but are there any ones that you think I could answer that would be useful? If you want to help, you know, with your microscope or anything like that too, I can help as well. So also I just forgot, but I have a whole bunch of guides that I were left in my Suburban that I brought out. You're free to look at them, pass them around. It's fine. Yes.
Yeah, on that page when it said 40 to 50 micrometers long, and the Catanella was like 40 wide, 18 to 44 between 40 long, 18 to 44 wide. So they're really about the same.
Yeah, because it's round, rather than that compression, I mean, you can kind of tell the size range it overlaps. Yeah, we see them a lot smaller though. I probably should maybe update that. Also, pretty much every one of my guides on harmful algal blooms covers Alexandrium because it's everywhere. And so there's all kinds of references in these guides in here.
I have a question, and I realize this could be coincidental. We had a large bloom of Alexandrium last summer on Karenia, the biggest roof, and it's not huge. And normally we see Noctiluca like bloom in August, nothing. I mean, not even one. Can they actually be exclusive? And it was during that Alexandrium bloom. Do things like that happen?
Oh yeah. So one of the reasons we think that they produce the toxin is to outcompete other things. It's a liliopathic, right?
So it could be why we didn't see any Noctiluca?
Yeah, sure.
I mean, who knows?
Noctiluca is it's phagotropic, so it goes into real big and just eats everything, right? And the reason why it's a problem is because all that gut material breaks down and creates this big ammonia and puts ammonia out there, which is why I see like a fish killer, right? Well, it's eating a whole bunch of these. It's probably got they're going to be affected by that like shellfish comes. So anything that grows is on this is going to be affected by the toxin.
I mean, we didn't even count one.
Yeah, there's a lot of seasonality too, and stuff like some years I just have the usual stuff that like, oh, I see this every year, and then other years like the last 2 years just saw a bunch of weird stuff that I never seen before. So don't get too accustomed to seasonality. It's 25 years of doing this.
Just have a look at the document and see if it, you know, what do you see?
Yeah. Yeah, it could is an example of things that change. This like that blue guide right there is I mean, the one that's in the photo. So that is a book of diatom descriptions put together by Esther Peck back in 1948 at University of Santa Cruz. All of that stuff in there is what we have up here. So we're seeing this big shift of diatom speciation and dinoflagellates also kind of moving from south to north. So since 1950, things have changed from being down there to being up there.
That's interesting. So, and are there things?
Yeah, well, that's the last few years I've been seeing all this stuff that is, supposed to be, out down off of Baja or, in Korea or Thailand or China, and now we're seeing all that stuff over here. All these species that are showing up. Yeah. Karenia, yeah,
Which ones are you seeing on the outer coast?
Just talking about that. So we sent her a bunch of samples and hopefully we'll get some genetic information on that to figure out what it is for sure. Morphologically,
Water, it's just outer coast.
so far it's just the outer coast. So, but there's a big gap between like Mia Bay and Port Angeles, which is the next, sample station. So, I mean, it can be out there, it can be in here.
A gap between different adjacent bays.
Yeah, it's hard to find it. Yeah, but yeah, and then actually, maybe tomorrow too, I have a list of all these, all of our Omphalogodon species that we know are here, but also there's a whole bunch of them that we just showed up in recent years as well.
And because of daughters and the big ones that it's out there.
There's a lot of work being done, so there's an increase in Omphalogodon's distribution and toxicity in relation to heat. And so if you are coming to water, and for whatever reason, the Omphalogodon species, especially the Alexandrium, and a lot of the general species, they do well with the heat. They can also live in a wide range of temperature and salinity, and they just generally repeat endemic non-species that are in the area. They'll just do better. So as things change, the HAB species do better.
These kinds of things related to climate change.
I could spend a whole day on
it.
I have a slide that my kids call the doom and gloom slide. It's like really depressing about how things have changed since the industrial age, but yeah, there's been a lot of work done, not my work, but there's
So you work your findings on
Vera, my boss, on a paper with Mark Wells and John Anderson and some other big names as well, looking at climate change and Omphalogodons. So
is that a recent?
I think it's like 3-4 years old. I mean, you should probably be able to Google Omphalogodons climate change, and that'd be one of the papers that came out. So, yeah, I mean, if you can still find that one.
I was hoping Vera was going to be here because, we have that photo of the polychaete that was eating Alexandrium, and it's just all along the digestive problem. The first thing people ask me when I show it to them is, did it kill the polychaete?
I mean, yeah.
I mean, they do have voltage-gated sodium channels.
Yeah,
so,
you would think,
but
yeah.
I saw an old paper by Vera's where the soft shelf clams have some mutations. I mean,
Yeah.
kind of intriguing, but probably not much work is being done along.
Yeah. I mean, I'd be worried about, polychaete worm like becoming a vector species, like, the bioaccumulation of the toxin. Every cell that it's eating, it's picking up that toxin, and something will eat that polychaete and get sick. So.
It was an aberrant polychaete.
Oh.
I did share the screen, but that's it. Also, that's my slide. I didn't your slide, but it's probably really great.
So,
yeah.
And then I think I'm good at. They often. That like. And so. No, not really. We never seen it. Not that I ever saw about how microsystem and that the structure of the top.
So somebody at our table asked how they get stuck together. Is that like, do they just not separate when they divide? And- Okay. Oh, it's not music.
I know that they water.
There's music which produced in response to certain conditions where. Okay.
But I know also the tomorrow that to get talked by. This is another way. But well, I think we have to balance, so you don't find. Yeah.
Where's the change the yellow of the leaf? So.
It just goes on the outside.
Okay.
Basically. It's
Okay.
So it's a small. Series of.
Still working.
So yeah. And then.
As far as I'm concerned.
I hate to say never. I told people never.
So I was
say
asking
never.
why they're stuck together. What? Doesn't mean word.
Yeah, I have like a big fancy picture when they just started. It's like 1 or 2% of them, but it's why it's not out for it.
Yes.
Yeah. And then it's
So, yes, there was
little.
and there were like little bees on a string, and I was wondering if they were playing with I'm happy if I just know it's laying the string. It was for my nephew. Michelle was just saying that, kind of wash it down.
Yeah. I was thinking this today.
Yeah.
Hi, everyone online. We are coming back from lunch, and we're gonna start with Azadinium. And I want to mention that this year we are going to be adding to our protocol Azadinium and Karenia as our new target or required species. Tracy put a note in the chat earlier too that says the National Shellfish Sanitation Program and FDA require that Washington has a contingency plan for monitoring NSP, which is neurotoxic shellfish poisoning, which can be caused by Karenia, and AZP, which is Azadinium shellfish, asparacid shellfish poisoning, named after Azadinium. And the Washington program uses phytoplankton monitoring to meet that requirement. So that's part of the reason why we're making them required species this year, because we know Azadinium's in our water. We did some, I think SoundToxins was an important part of identifying a few species of this for a research project, I think in 2014 time period. So some of you might remember that. I do not, because I wasn't here. But if you do, we know that they're in the water here. So we're looking. We want to kind of focus our efforts on looking for them. And then again, the Karenia ORHAB saw on the coast this past summer. So now we're kind of we knew it was always a potential, but now we're kind of a little bit more on high alert. So so we're going to start off after lunch with Azadinium, and Anthony is going to start with the presentation, and then we have some pictures that we're going to pass around for folks to see. So here is Anthony. And here is the slide that I'm going to share. All right. Oops. There you
go. Okay. So Azadinium, if you guys aren't aware, is incredibly small. If you're looking at a preserved sample, you might not catch it. It's kind of this little acorn shape with this really broad cingulum, and the cingulum compared to the rest of the theca is kind of what can give it away. It does look like a very small Heterosigma or Cryptidinium. So there's some confusion. Lots of times people think they see this, and it's actually Cryptidinium or Heterocapsis, sorry, and Cryptidinium. So it looks very much like that. But how you can tell is by, come on, play. You gonna do it? I see I can't get it to do it on your computer, but I can get it to do on mine. No. I might have to show from my computer. Anyway, we can talk about it real quick. It's dorsal-ventrally compressed, which is actually nice that if you do get one to sit still, you can see that very narrow sulcus and very wide cingulum. Again, this is another reason why I need to be looking at live samples after you collect them. Once you see this swimming pattern, you'll recognize it when you see it again. They'll spin in this little kind of circle, and then they'll jut off at an angle, and then they'll spin again, and they'll jut off at an angle. And we have this video, but also what's that? Oh, I got. I think you can sign back in. So yeah, Nick Adams in 2017, he did some survey work and used PCR that showed that we have Azadinium spinosum, paporum, and abyssum here. But there's very well could be other species here present in Puget Sound and on the west coast of Washington. There's a link down there to at the very bottom to algae base. Also, again, if you guys have attended any of my Zooms, you know that I'm going to really hit hard with the algae base. It's not always 100%, but it's fairly up to date. It will, if you find something and you want to confirm it, go to algae base. You think that you see it, look up the name at the top, and it will tell you if the name has changed. It will give you all the literature. It'll tell you known distribution, maybe not entirely accurate distribution because you're always kind of behind on that. But it'll also, you'll see it in my talks. I quote it a lot too. It's not the final authority because of some of the stuff in algae-based people are still kind of arguing over, and they'll put it in algae-based and they're like, is that true or not? So you'll see some change going back and forth. That's just taxonomy. It's how that works. But yeah, if we can get it.
Type in your password.
Do I have to? Okay.
Sorry.
I'm going to switch over to the other computer and then we'll watch this video.
We'll try to get the video in 1 second. Oh, it's working.
Well, now it's working. You had to touch it.
Sorry, yeah.
No, it's fine.
My computer is like bio-signified to me. Only response. Okay, there you go. You want to talk about
no, I mean, that's it. So yeah, then we'll do a little bit of close-up. You can see it has a really easily identifiable swimming pattern. Where it'll just be kind of like lazy, then it'll do a little spin and then it'll just take off. Can you get it to do it again?
Yes, I can.
I'm not touching. Yeah, what's with that? Okay.
You see these spin.
So it's good to see it in culture because you can see this pattern all over. But if you're looking at a wild sample, you be like, that's Azadinium, because it'll bing, bing. So yes. So we're gonna look at some samples. We had some sent from Germany that Brian Bill at NOAA graciously brought back to life.
By way of Massachusetts.
Yeah, by way of Massachusetts, yeah. So yeah, it's a really good sample. It's pretty loaded. I do have it on computer, but the Zen software that I use for my camera is competing with the zoom on the video card. So it is a little herky-jerky, so we're not going to see them swim as well, but I can also zoom in very close so we can look at the morphology. So yeah. Yeah. Which is huge compared, right, to the bodies.
Right. This is the nucleus.
Yeah.
Is this the pustule?
Yeah. Yeah.
The pustule.
Yeah.
Yeah. The nucleus is this.
Yes.
It's okay if I jump in. So I was taught that for dinoflagellates, one way to identify the nucleus is to look for something that might look like a thumbprint. So if you see like I don't know, like ridges on it.
See the chromosomes, they're really organized and they're really thick. And so it will look like a thumbprint. You'll see like these just series of little lines going up.
Yeah. So something that looks like, yeah, like a fingerprint or a thumbprint. And then if something looks like perfectly round, that's usually a pustule.
Yeah.
And kind of smaller in comparison.
Yeah.
What does a pustule do? Does anyone know off the top of their head? Or else maybe I'll Google it and tell you. It's a type of it's an organelle, but I don't know exactly what it's for.
Yeah, you can Google it.
I'll Google
I
it.
don't want to be wrong.
Okay. Give me a couple minutes on that one.
Yeah. Yeah, so I'll sign in here and I have the microscope hooked up and we can look it up here, but also there's two flasks here. One of them, this is kind of the original one and it looks very thick, but I think there's a lot of bacteria in there. This is the one that Ryan brought back to life. It's very clean. Doesn't look like there's much in it, but it's lovely. So
is that the one you used for your sample? Okay, so maybe we'll just use that one then.
Sure.
Can you do that, Melissa? Thank you.
Okay.
Okay, so yeah, in a couple, in a minute or two here for the folks online, we'll get some live samples up. I'll play the video again while waiting.
Yes.
Anthony has both of them, and he's looking at one now, but I guess get ready for the other one. Either of them should be fine.
Do you want to do the legals? And I'll do
Oh, yeah, I'll do the legals. Perfect. Oh, great. They were next to each other in the slideshow. So that works.
Should I give them out now or wait?
Let's wait. Okay. Okay. Okay, we're gonna move on. Here's Anthony to talk about Heterosigma.
You guys all have Azadinium down. You saw the swimming pattern, right? Very small. Look at live samples. So this is Heterosigma Akashiwo. It is a common rhophytophyte. It is a desmocont dinoflagellate, meaning that it has two flagellets extending from a central groove up near the apex. In the case of many rhophytophytes, but especially Heterosigma, the leading flagella actually pulls it along. It has, if you get really close, which you probably can't see on a light microscope, it has little hairs on it. That leading flagella is called a flimmer, and it pulls it along, and it does most of the work, and then the trailing flagella, it kind of does a little bit of work. It kind of just barely. You'll see from these photos that there is a high degree of morphological variability. You can see it in all these forms. The sample that we're going to look at, but you can see that there's many different forms. It's basically just a bag of chloroplasts, and the chloroplasts are kind of this rusty, kind of orange brown to yellow. Freshwater rhophytophytes can have a green, but these ones are, yeah, kind of this orange color. If you put lugalls on them, they shrink up and they kind of look like a little raspberry or a lumpy sack of potatoes. Lugalls preserved samples is kind of the way to go with most aficated or naked or thinly fecated flagellates. If you put formalin on them, especially if it's doesn't have the right pH, it'll make them pop. So lugalls you kind of have to tend to a lot more, and you have to kind of go back and check and keep adding lugalls every few months. That's not like a formalin that just kind of preserves things kind of indefinitely, but it will keep these things together. And so that's one of the reasons why we're going to look at samples that have lugalls on them as well. So yeah, again, this is like one of my least favorite taxonomical terms, morphologically variable. Which, yeah, it doesn't really help on light microscope, but you're going to see swimming pattern on here as well. And really nothing in our region really looks like this or has that color. And it's just a matter of seeing enough of them that you'll start to recognize them. If you have seen a bloom of them, you know that they turn the water kind of this bright orange color if they're really thick. But again, this is why we're looking at culture, so you can see them in kind of all of their morphologies. These are big fish killer. It's ichthyotoxic, and they're present everywhere, pretty much everywhere, and cosmopolitan in temperate waters for the most part. Coastal and brackish, they like embayments. Because they're naked flagellate, they kind of like shallow retentive areas where they can vertically migrate, so they kind of will go up and down through the water column. As I was telling Michelle earlier, I was doing a training like 2008 or 2009, and Brian Bill, the same gentleman who brought us these cultures today, he gave me a culture of Heterosigma, and I had it in a scintillation vial, and I just put it on my windowsill by my desk, and every morning when I came to work, I could watch it vertically migrate and kind of come up from the bottom and a little later, and then in the afternoon it kind of dissipate and go down to the bottom. I did that for about 3 years. I never even like took the cap off. I just like lived in this scintillation vial, and then one day it was just like, oh, you died. Yeah, so they're sturdy, they're hardy. But that's also another way to see if you have it, because they're such strong vertical migrators. If you just like leave it in like kind of low light, maybe uncap the lid so they get a little air, and everything else be settled at the bottom. If you see like a little layer coming up, you're like, oh, I probably have Heterosigma there. Yeah. And oh yes.
So I think Anthony mentioned that they are morphologically variable, and there has been a kind of like a weird mutation of them that has been seen specifically in East Sound a couple of times. And I don't know if anyone else has seen them, but we've been calling them the Pac-Man ones because they kind of warp and look almost like kind of like a Pac-Man, but also kind of just like kind of wobbly on the edges. So I think we need to get a picture of that shared, as part of the slide, so you can just see like kind of how different they can look. But again, if you see something weird and you see a lot of them and like, you're like, what is this? Send us a picture. And we might be like, we have no idea what that is, or we might be like, one time we saw something that looked similar to this and it was heterosigma. So, oh yeah, okay, so maybe we can share that picture just in case. And I don't know, I don't know why they do that morphology. I don't know if it's something in the environment or whatever, but it seems that it's happened at least a couple of times in the same area. But you've also seen it in its other morphologies, like more. So maybe when it gets like a super thick bloom, it can like change a little bit. But yeah, so anyways, just they can be weird.
Yeah, I mean, if you're just going by all these pictures up here,
yeah, they all look
there's
kind of
several morphologies, right? And even like the line drawings that are in the same manual, like they don't look anything alike, right? So yeah, anyway, we're gonna look at a sample now, gonna get some Lugol's on it. You can kind of see it in both of its forms.
Yeah. And then for the folks online, Anthony, first you're going to do the live one, and then we'll give you a Lugol's one as well to see.
Yes.
Okay.
Yeah.
So I'll try to drop in the chat when we like add the Lugol's, so it might be obvious, but just in case it isn't, we'll try to mark that. Okay, we're gonna come around. Melissa's coming around with that, and then I'm gonna get some Lugol's and come around after. Okay, great. Okay, everyone. We're gonna do one more culture before our afternoon break, and then we have a couple of species after the break, and then we'll have plenty of time to look at our wild stuff that you guys have. And also, if you want to go back and look at anything, there'll be a bit of time for that, because Anthony noted that in the Alexandrium samples, there are several different morphotypes. And so if you want to go back and look at that one afterwards, and we can kind of, you know, just get a little bit more practice with that and seeing the different morphotypes, we'll have time for that. Okay, so the next one is going to be Pseudo-nitzschia. And I will share the screen. No, I can take over, since I am the owner of this meeting. Okay, here we go. Share, replace current share with PowerPoint. All right, here we go. Okay, here we go. That's yours.
Okay, so we're gonna look at a sample, a culture of Pseudo-nitzschia pungens, which is a known toxin producer. As I go through these, I want to note that everything that you see here, both with light microscope and with scanning electron microscope, everything is a different species. So similar as those all look, they're different. Out here, we kind of group them as large and small, and I'll kind of talk about what that means. And a lot of these descriptions, they will talk about length, width, and cell overlap. So Pseudo-nitzschia form these chains that have cell overlap. They kind of look like a grain of rice. And they have like a pointed, blunted or rounded ends, but they generally taper for the most part. They have two chloroplasts that are generally center with a gap in between them. On some species, you can actually see this structure here. So the only way to truly tell all of the different species apart is right here is a raphe. A raphe is like a longitudinal slit that goes down the length of the diatom that's held together by fibulae, little ribs on the inside of the structure. That's this part right here. That raphe is open to the outside. It allows for exchange of materials, but also you will see sometimes on Pseudo-nitzschia on your slide, you'll see it moving along. Okay, it's ejecting material to move itself along, and it's basically when it comes in contact with something, it wants to get floating again. If it's free floating, you're not going to really see it move, but if it's touching something, your glass slide, you're going to see it move. But that comes out of the raphe. If you want more information on that, it's pretty complex. I can get into it if you want. But how you identify the different species is these are pores or poroids, and this is the striae, and this is the interstriae. And you have to look at these rows of poroids in the striae and also in relation to the fibulae, and you have to do some counting and you know, how many striae per fibulae, you know, what the poroids look like on the inside, how many there are. Can't do that with a light microscope. So the whole takeaway with this is you cannot tell Pseudo-nitzschia to species using a light microscope. Now, no matter how good you think you are, I was doing this for years and really messed up one time when we had a big bloom of Pseudo-nitzschia australis, which is like the most toxic of the genus, and thought it was Pseudo-nitzschia pungens because of its size. And so I saw it, I'm like, yeah, there's a lot of it, but it's, you know, not really known to make toxin. If it does, it happens at the end of the bloom and it'll produce a, you know, a little Alexandrium, I'm sorry, Pseudo-nitzschia australis, like front loads domoic acid, just puts a whole bunch out there all the time. Clams became instantly toxic. And so I was very wrong. And it happened in a matter of days. So that was like the lesson I already been told, like, if you look on a light microscope, you can't tell. And I'm like, but I've been doing this a long time. No, you can't tell. So yeah, lesson learned the hard way. Here's kind of an example of what I just went through. The different parts of the cell. Again, can't see in a light microscope, but I'm going to go through this really quick. Again, this is kind of explaining the raphe slit and the fibulae. Oftentimes along that Rafe slit, there will be something called an interspace, like it's an extra large gap in the fibulae. It's like central on the cell. When you're doing SEM or something, you know, the presence or absence of that interspace can help you identify the cell as well. Again, everything I was just talking about again. This picture down here is one that I took in 2015. This is off the Santa Barbara. 2015, we had the big bloom of Pseudo-nitzschia. We had tons of marine mammal mortalities. At the time, it was the biggest harmful algal bloom ever. It extended from the Santa Barbara Channel all the way up to at least southern, like BC, but probably up into Alaska as well. We had large die-offs of fin whales. We had 6 seals, lions, birds, kind of everything. I was actually out at sea for this and we were doing a grid up and down the coast. And as you can see, this looks like it was grown in a lab. It was a monospecific bloom of Pseudo-nitzschia australis that extended from the near shore as far as we looked out 200 and, to 210 nautical miles offshore. It was completely through the whole photic zone. And then below the photic zone, it was raining down empty frustrals as far as we could see. This is using the acoustics on the ship. That was all there was from Santa Barbara all the way up to Washington. From the coast as far as we look, there were gray whales and humpback whales were going into rivers to escape it. You couldn't eat anything. We would see large kind of pods of sea lions and they'd be in the water and very sick, intoxicated. This is really highly toxic. Domoic acid causes amnesic shellfish poisoning. And so if it doesn't kill you, it can give you permanent short-term memory loss. But it has a wide range of symptoms. Flu-like at first, neurological, people can develop stutters, they can develop mutism, they can develop hiccups that don't go away. So there's a wide range of gastrointestinal to neurological stuff that will happen before you get to the point where you lose your ability to put short-term memory into long-term memory. So if any guys have seen the movie 50 First Dates, where she hits her head and thinks every day it's the same day, that's what happens. There, in the original outbreak in 1986, where this domoic acid was isolated in Prince Edward Island, one of the gentlemen that got sick was a physics instructor, and he could remember his talks because they were in his long-term memory, but he couldn't make new memories. So like his wife had to tell him every day, like, you lost your memory several months ago. He eventually passed away. Not too long after this. Several people fell ill in that bloom. And so they were able to determine that everybody pretty much had shellfish, and most of them had eaten mussels at some point in the last few days. And like all of the hospitals, clinics, everything was filling up all at once. And they were able to isolate domoic acid. And then since then, the more we look, the more we're seeing it. We're also seeing that it goes back in the taxonomic records when we look, that it's actually been around for a long time. And it could be that people have been getting sick all along, but if you don't know a causative agent, you're not going to, really understand. You ate some bad shellfish. In our case, out on like the Washington coast, if you're out digging razor clams in the winter, and when you're done, people have flu-like symptoms, you're like, well, you've been out in the elements. You've been in the weather, it's bad, that's why you don't feel good. If grandma starts losing her memory or little Johnny develops learning disabilities, you're not going to relate it to whatever it was the razor clams were eating if you don't understand that it's there. So it could be that this stuff has been around for like a long time and people just didn't recognize it. It's still a bit of a problem in that it's hard to get doctors and nurses and people in clinics, especially out on the coast, to kind of remember that this stuff is out there. And so, a big thing people say is like you're having allergic reaction to shellfish. It could be domoic acid, it could be diuretic, shellfish toxins, Alexandrium. Alexandriums are like a little bit better known, but this starts off with flu-like symptoms. So those are kind of the issues with Pseudo-nitzschia and domoic acid, amnesic shellfish poisoning. It's why we need to monitor for it and we need to look for it. Some of the cells like Pseudo-nitzschia australis, that species, we set an action limit at 30,000 cells per liter, because at that point, it's known to make enough toxin where you see uptake in shellfish. So that's actually a pretty low number. So that's our action level. But then you have other species like pungents in multi-series that don't produce a lot of toxin, or they wait till they're stressed and they're getting grazed on, or they're dying off, and so they'll produce it at the end of the bloom, and it can take 150,000 to 250,000 cells per liter. So because of this, we have this really low action level. So we get like event response mode that'll last for months because we're not really sure what the species is. And then even when you think you're confident of the species, you're like, okay, it's not Australis. You have Pseudo-nitzschia pungens blooming, for instance. You can have Australis start to come into the bloom and you can't tell the two apart. So you have to be in event response mode like the whole time, just when you see it. So there's some key to species down at the bottom. It's prevalent. There's been a lot of work that's been done on it, a lot in our region. It's actually how my program kind of started, like why I'm here. So, so this is what we would call small. We have Pseudo-nitzschia delicatisma, Pseudo-delicatisma cuspidata, and a few others that are this small. And you can see they're very, very thin, and they are in relation. If you can see the large cell and the small cell together, you can see the size difference is noticeable. But you can barely see the two chloroplasts in each cell. They're very narrow. They don't have a whole lot of overlap, 1/8 to 1/9 cell length. People always ask me, how can you tell large and small? And honestly, you really kind of need to see them together. The cell range on this one makes it really frustrating, 50 to 140 micrometers long. So they're like maybe cell width, but also 1.5 to 3.4 micrometers wide. That falls into the range of large, right? So it can be difficult. Just you can put CF, what you think it is, CF small even. But you can't, without SEM, you can't tell the difference. This is delicatisma. It's a little bit broader, but if you look at the range of that one, like these cells down here are raised in culture, I think that's why they're so thick. The cell range is not quite as much, but also you really can't tell the two. I mean, this is the whole takeaway I want from this is Pseudo-nitzschia. Recognize that it's this forms chains that are overlapping. You see one, again, like I said before with Alexandrium and Lurbitz, not every pennate diatom is Pseudo-nitzschia. There's lots of pennate diatoms. So yeah, it helps if you see it forming a chain. It's the only one that has that overlaps. So Australis, this is really the nasty one. You can see it generally is a little bit broader. It has a pretty good size range as well. Fraudulenta looks like almost identical to Australis, not nearly as toxic. There's pungens. This is what we're going to look at today. Again, this falls in the range of Australis again. Yeah, we had this big bloom all last summer. We were getting into the millions of cells per liter of pungens, and it never produced toxin. Not the whole time. Like domoic acid levels are still fine in razor clams. We saw like one little bump, and then that was it. So right as it was dying off. Multi-series looks just like pungens. Yeah, and that's it. So let's look at this sample. But yeah, that's the takeaway of Pseudo-nitzschia is you can't tell species light microscope. Just treat it like it's the most toxic kind. Anything to add?
Yeah, I guess I'll just add on to that in our inland waters, it very frequently does not produce toxin, but it has historically in the past a couple of times. And so it's kind of what Anthony was saying, like kind of always treat it like it's dangerous, even though it kind of feels like you're reporting it all the time and nothing happens. Checked in with Tracy and Jerry at DOH, and they said, yes, just keep letting us know when you're seeing it blooming, because we don't really know when it's gonna pop up and become toxic. So continue to let us know when you see it. Yeah, that's all.
Any questions on Pseudo-nitzschia? I think I might be able to answer. Let's look at some samples.
Awesome.
Also,
Okay.
I have a list of symptoms, and I will share that in a minute because it's wild. Like the having hiccups that don't go away. Like I can't have hiccups for 3 minutes without being like, losing my mind, but go for like a day or something. Yeah,
Yeah, really terrible. It's terrible. Hiccups that don't go away. Oh.
Right? Yeah.
Yeah, okay, so I think we have one culture today, and I think it's this one. Oh, one other note, Pseudo-nitzschia can, a lot of these cultures, in case we didn't mention it, might look different when they're in culture versus when you see them out in the wild. So when these cultures that we're getting, they've been given like really high nutrient medium to grow in, and so it's not the same conditions that they would be having out in the wild where they're competing for all these other organisms for all the nutrients and everything. So it sometimes causes morphological variabilities when you're looking at them in culture versus in the wild, which is frustrating considering this is our like best chance to see them, you know. So these ones I think Brian mentioned might be a little bumpy, so not like as perfectly smooth as you normally see them. So we'll take a look at them. I haven't actually looked at them yet, so we'll see what they look like. But if you're seeing things like bumps and stuff on the edge, that's not necessarily characteristic of these. It's more just like a weird thing that happened because they're in culture. So yeah, I think that's all I have to share. And then after this, we'll have a little break with a couple of snacks. So.
And what? Chain formation? Like one way to tell them apart.
Oh, yeah. Oh, yeah. And Kate also knows a lot about Pseudo-nitzschia, so please help us. Help us.
I just wanted to mention something that helps me differentiate the pungens and multi-series types from the Australis seriata fraudulenta types, just because I think it's helpful. I'm gonna see. But basically for Australis seriata fraudulenta, those are wider cells, and so that's something to keep a close watch on. And the other thing is that when you look at the chains themselves, you'll be looking at Pseudo-nitzschia pungens. Pungens and multi-series, they tend to be a little pointier on the tips, but they also make the stair colonies. So the cells line up almost like a stairway. And for Australis and seriata, you see almost a straight line on one side and scalloped on the other side. And so that's just one way. Again, you have to make sure that you're in the right view to be able to see that, so you can still get them confused. But it's one of those things that I really, trying to figure these out, it's always a pain. And pungens and multi-series are a little bit thinner. And another trick that I try to do is the cells can only be up to 9 microns wide. And so when you think about trying to compare the length versus the width, the little bitty guys are about a micron wide, the big ones are about 9 microns wide. And so sometimes you can just use that to kind of help size things in your head. But that scalloping is really helpful. And Brian Bell taught me that a long time ago, so I'm just regurgitating his good words.
Another shout out to Brian. And also as a sneak preview, I think Kate is going to talk a little bit about the times that Pseudo-nitzschia got toxic in the sounds tomorrow. Is that true? Okay, great. So that will be kind of cool. Thank you. Okay, are you gonna? Yep, I'm gonna stop the share and. Are they? Okay, I'm hearing that these are a mess. These might not be the best example of these, but you know what? It's something. It's the sample. Yeah, I can see this.
too.
Okay. So these cells are kind of bent in themselves, and you'll see that central interspace between the chloroplasts, there'll be like a little bulge, and that's causing the cells to bend, and that's why they're making these kind of curved change. That's not something you would see in nature. They would be straight. They wouldn't, these look, they're a little rough, and again, it's because they're getting fed medium. So they're extra happy, they're growing really fast, and they're doing weird Frankenstein stuff, so.
But we also just heard that some of the wild samples that people brought in today might have Pseudo-nitzschia in them. Some of them definitely do, some of them might. So we'll get a chance to see what they look like in the wild later this afternoon.
Does anybody want to use the magic filter? Yeah, okay, there we go.
Yeah, especially if that's going to be easier for you to add than like going in and changing like the drop down and the thing. I think that's great. If I show up anything, they shut anything down. I want to actually like. Okay, but this is also like kind of a weird thing. We're re-examining our protocols and trying to look at like what it is that's good. Different I want to have a grad there. One of of like I know. Right, but that one is just like slow count. So there might be something we can do in there. And then the other thing is, in theory, the whole water like sounds more accurate. So we going to so depending on like how different buckets might be, you know, like if yeah. Well, let's we're gonna have a community discussion session tomorrow, and one thing that we're planning to do is get a like re-examine like what we do, why we do it. We like really want people like people are doing it, input, especially someone who's been doing it for like a long time. And I'm wondering, like, but then also, like, why aren't touching on the net? Does that mean, like, when you're pulling it, and somehow or contributing? Yeah, I mean, like, it's a whole water. Well, that's the thing about the whole or the net code calculation is like I think the theory behind it is good, but there's so many variables that are different and like how you pull the net and if there's flushing and like rinsing the net that like factor into like how much treatment comes those. I don't want to waste everyone's time with the students been doing, so I'm like they keep doing it, but I think now's a good time to like kind of re-examine the protocols and get like more experts in the room and try to figure out a way that can be efficient for folks that are doing everything, but also give us like the information that's actually like because I think the information that we have now is like good, but could it be better and like yeah, the best way to capture everything that's in the water column without looking at like water. You don't want that either. Yeah.
Is that a question if they're like, yeah, do they connect top regions or is it like they're like, are they dividing? You know, it's like they're dividing, I think they just like
yeah. Yeah. How do they? That's a good question.
Wherever you're showing the we will sketch, then it looks like they're like moving apart.
Oh, are they deciding again? So they're
like you can see it on our slide too. But if it's
special tenure. Yeah. Well, the seagulls.
Unmute video. Hello, everyone. Hopefully you all got snacks and are ready for more stuff. In case you didn't hear, Anthony put up this symptoms of domoic acid or amnesic shellfish poisoning, starting from like least concerning to most concerning, lots of different things here. So hopefully you got a chance to look at that. And then the next thing we're going to work on, do you want to talk about that at all, or you haven't already did?
Yeah.
Okay, great. Ask us more about this later if you want more information. And now we're going to work, we're going to take a look at our last culture, which is going to be a mixed group of Dinophysis. So I will take over the screen share. 1 second. Use current share. Okay, here we go. All right, go ahead.
Okay, so the next culture we have to look at is Dinophysis. And there's, she said it right in Dinophysis group. Species, maybe different species, maybe all the same species. Genetically, they're all very, very similar, and there's morphological variability from area to area. You're going to see in the culture we're looking at, there's a lot of overlap. What I believe it's Dinophysis acuminata, norvegica, and acuta, which in their morphologies, there is overlap. When I took the IOC HAB course in 2008 in Denmark, as part of the test, we're supposed to go through each sample and identify what was in there. As a part of the test, they did just that, where they put these 3 in one sample and were like, tell us what it is. And you could watch everybody's head explode because we were just, and I went up and I was like, well, there's 3 in there. And I wrote that down. They're like, how did you know the Dinophysis? I'm like, because it covered the whole range of morphologies that had to be at least 2, but probably 3. Yeah, I mean, I just kind of went for it, right? So, but yeah, it was pretty complex. The Dinophysis by itself is, you can tell to genus what it is. It's very large cell. It's very ornate. It basically has, it's called their singular lists, which looks like a crown on top. And then it has a sulcal list, which is, it's on the front side, the ventral side, but it kind of looks like a big dorsal fin, basically. And very large body. As far as telling the identification between the species, we look at size and shape. Some are whitest, like median, post-median. Some of them have like a rounded posterior end. Some of them come to a point. They're convex. You can also look at the sulcal list. It has little spines that kind of hold it out. And a sulcal list, again, so you have the sulcus and you have a trailing flagella, and the sulcal list protects that. And so there's a flagella, and there's actually a left one and a right one. And one is larger and one is smaller. So, and then there's a flagella that goes down in between of them. Same with the singular list, which is this crown. So you have a cingulum that is, it's almost apical. It's on the very, very top. So it's very pre-median. And there's a flagella in there. And then it has this huge singular list that stick out, that has ribs that hold it out, that also protects that flagella. So having the transverse flagella and the cingulum at the apex, it's really good steering mechanism. You have your steering flagella like right up front, and then the trailing flagella goes the whole length of the body and is wiggling inside that sulcal list. And so you also have good propulsion. So I mean, it's actually a pretty smart design, especially if you're heterotrophic or mixotrophic, like you like to eat other things. This thing can steer really well, and it's fast, and it's large. Also, we look at the antipical or posterior end to see if there's spines or horns, absence or presence of chloroplasts. Now, these aren't born with chloroplasts. They steal them from other things. So it's kleptoplasty. So these are kleptochloroplasts. They steal them from diatoms. So, and if you can think of it like if they ate the chloroplast, that's like eating the donut. If they steal it, put it in their body, now they have a donut factory, right? So it's making lipids and sugars, complex carbohydrates, and it's so, and they have that all of the time. And then if there's low food, they just have this food factory in them. Pretty smart, pretty ingenious. down the last note, species concept of Dinophysis and phalachroma is weak. Many species are poorly described. Taxonomic revisions are needed. And this comes from like the horse's mouth. This is like the the best dinoflagellate guys and Jakob Larsen, who specializes in this in the University of Copenhagen, I believe. Like Dinophysis is his bug, and he's like, it's a mess. And when they do all the taxonomy, they're so close genetically that you're kind of like, is it really a different species or just morphological variation? And are there like cryptic species as well? And we just assume that it's this because it looks like that. So the more we look around the world, we're finding that it's not as easy as morphology. Again, Dinophysis, CF, what do you think it looks like? And so we're going to go with some basic morphologies, but just so you know, again, the more we look in taxonomy and the more genetics comes into play, and we're kind of moving away from morphology, the more complicated it gets. So in general, this is just a drawing and kind of pointed out all those things. So the ventral side is where that sulcal list is. So it's that's the belly, that's the front. The posterior side is the round side on the back that has generally no ornamentation on it. The cingulum or the girdle is that crown up at the top. It has a left and right sulcal list. Really, you can't tell the difference between the two, even when they're so close together that when you're adjusting your focal plane and out, you generally only see the larger sulcal list. Cell length is the apex to the antepex or anterior to posterior end. The midline is the widest point. And so we'll say, and that's also like where, the midline is kind of a general anterior to posterior line. They're not symmetrical ventral to dorsal, but so you just kind of like guess this is kind of in the middle. And the reason why that's important is where the tip is, if it's beyond that or forward of that. And then cell width just means at its widest point. And so also cell width can be like widest median, post-median, pre-median. So if you kind of remember those terms about where it is on the cell as well, that's going to help us identify different Dinophysis species. I'm not going to go through each description because I kind of want to get on the microscopes.
Oh, I just want to update that.
Oh yeah.
I just checked the notes and the mixture is 40i, which is this one,
Okay.
Norvegica and Acuminata.
Okay.
And there might be some Azadinium rubrum in the culture because that's what they were mentioned on.
So 40i is actually easiest to tell apart. It's widest post-median, has a big belly. And it's a fairly large cell. It's medium for Dinophysis. It's probably one of the more easiest to identify within the genus. Around the world, in other places in the world, this is the most toxic strain. Ours is Acuminata regionally. Also, if you want papers on any of the stuff I'm talking about today, I can direct you towards that. Where this resides, Neretic and oceanic, so nearshore, offshore, cold, temperate to tropical waters worldwide. That's like everywhere, right? And you find this up in Alaska and in the Bering and in the North Atlantic as well. So it has a wide range. Tropical. In Japanese waters, this is the most toxic, and on the Western Pacific, it is generally the most toxic. Acuminata, again, morphologically variable, favorite term. Sometimes you'll see it with these little bumps on the edge. Those are called bosses. In some of the original descriptions, bosses were deterministic of species. Now we know that it's probably just a factor of age. So those little bumps on the bottom that can't really tell you the species. Norvegica also gets these as well. This is why there was some confusion in which one's which. But you can see they can be ovate to somewhat pointed. They're fairly long and skinny. If you look at the singular list, in a lot of these or most of these, that cingulum is so wide that on the posterior singular list, you can actually almost look inside of it and it's not really hiding the flagella. And that anterior singular list is like, that crown sits very, very high on its head, basically. Acuta. This one also is also very, very common. Acuta, it has an acute posterior end, right? So it kind of, it's kind of concave somewhat underneath the sulcal list, but that's how I remember it. Also, those little bumps, it's areolation, strongly areolate. Some of those are just kind of like cups or pores, and some of them actually go through. Just allows for the passage of the material from the water into the body and vice versa, waste out. Also, as we're looking through the culture, if you guys have more questions about it, you can come to me. I could spend really all day on Dinophysis and Phallochroma. This is Norvegica, and if you look again at the line drawings and the photos, there's a lot of variability. And if I go back to Acuminata, like if these were in the same sample, you know, and then also, like I said, as the more we look, we're realizing genetically they're so similar that it might just be variation, and might have to come up with a new name altogether and say these are the variants of it. And again, there's probably a lot of cryptic species, things that we just are assuming based on morphology, this is what it is, and then we do the metabarcoding, and it's like way different than what we're calling it in someplace else. So a little more complex. I'm glad Jakob is doing all this work and not me. Dinophysis caudata. So this is another one. This is actually Dinophysis tripos var variety caudata. Genetically, it is identical to this one. So, but we see both here. So you know, one horn or two, kind of camels, you know, one lump or two. We see both here, but yeah, or cut out of our tripos. Sorry, I got that backwards. So this is the type species. This is a variation. But yeah, we see both. I mean, with that posterior end going to that horn, that point, that big projection, that's obvious. You know, it's one of those two, right? Dinophysis parva, generally smaller. It's widest pre-median, so it kind of looks like it has its chest puffed out, like has little man syndrome, right? It's like this little Dinophysis and it's got its chest puffed out like it's ready to go. Has a very small sulcal list. Yeah, you can kind of, for the most part, go off of morphology with a lot of these. Okay, now this is Phallochroma rotundatum. So Phallochroma looks like Dinophysis, genetically very different. Not helpful. How you can tell Phallochroma from Dinophysis is if you look at the singular list, that crown, with Phallochroma, you can see that bald head above the crown, okay? So if you can see the apex above the singular list, that's Phallochroma. If you can't see it, that's Dinophysis. If that crown sits high, it's Dinophysis. So that's how you're going to tell. Also, there's only a few of these formerly Dinophysis species that were pushed into Phallochroma. Also, are you guys aware of Oxyphysis oxytoxoides? It is now Phallochroma. Because genetically, it's, even though they don't look anything alike, genetically they're like this close. Yeah. Convergent evolution, divergent evolution, like it's, yeah. Does that help with the Phallochroma versus Dinophysis though? When you look at the singular list, if you can see the bald head above the crown, it's Phallochroma. Unless it's the former Oxyphysis, which looks nothing like this, right? Yeah, is it pointing?
They're related, even though it's frustrating that they take.
Yeah, okay, close. Anyway, that's all I'm going to cover. I really wanted to get to look at the sample. And I really want you to see the difference, try to find the difference between Acuminata and Norvegica. And if you can do that, you move from beginner to intermediate. In 40i, everybody should be able to tell. So
Do you have a slide on Azadinium? I don't think so, right?
on what?
Azadinium?
No, I don't have. Yeah. Oh, so that's the other thing. Mesodynium is a little ciliate. It's kind of light bulb shaped, and it has a bunch of cilia going off each side. Turns the water bright red. Like one of the calls that I get the most, like the water's red, what's in it? It's Mesodynium. It's a harmless, benign ciliate. But if you see Mesodynium, there's a good chance that there's Dinophysis in the water because it's Dinophysis's favorite food. Brian was one of the ones to figure this out with some other people, that it was having a really hard time culturing Dinophysis. So they figured out what it eats, and they saw that when it bloomed in nature, Mesodynium was abundant. So they started giving it Mesodynium. And then it was like, wow, this is great. And they were able to keep it alive in culture. Like, we're doing it. Well, then they had to feed the Mesodynium little cryptophytes. And so there's like this layered, to keep it in culture, but they do it. And like, yeah, he has 3 different types here that he's growing. So, but yeah, I think that's it.
Awesome. Okay, we will pass out some samples now. And hopefully you'll be able to see all 4 species in here.
So there's one from like February, like the same thing, the one from February. Similar, but maybe not. The February one. Good to know it's more than the January. I see stuff in the February one too. Let's do it first and then we'll start over.
There's been a lot more now.
Okay.
There are.
Okay, thank you.
And I built more than.
This is like your first time. I know this is really overwhelming. The learning curve for phytoplankton taxonomy, you start like this, okay? And then you, if you know the Dunning-Kruger effect, you kind of hit this slope of despair for a while. You're like, this is too much, I'm never gonna learn. And then, then there's a slope of enlightenment. It takes years. I've been doing this for 25 years. There's still stuff that I don't know. I just went to this course that was 3.5 weeks with really people came out of retirement to teach this thing. I asked them questions. They're like, I have no idea. Like that'd be a great paper. Like, you should look into that. And the phytoplankton world is huge. There's hundreds of thousands of species. I don't have this stuff memorized. Like, I have to go in the book, and it's so broad and varied that I'm in the literature all the time. Don't get overwhelmed. Even the world's best are like, okay, so yeah, don't get discouraged.
I also want to prepare you all for some potentially emotional turmoil. For all of those of you who have learned all these species names and you're like, yes, I'm finally learning, I got it. And then they went and changed all the names. That is definitely pretty frustrating and emotional for some of us. So someone just came up and said, is this Oxyphysis really changed its name? And unfortunately, yes. Anthony and I spent a bit of time going through our species list and updating it, and some of them really hurt. I'm not gonna lie. So we will be sharing that information with you tomorrow. If you want to, in your heart and soul, keep calling it the old thing, that's fine. However, to keep up with the scientific literature and everything, we'll probably be doing some updates. We haven't like officially done it in the database yet, but I'm thinking about doing something like putting the new name and then saying like formally the other name, at least for like a couple years or something until we've all like learned the new name or like assimilated the two. It's definitely challenging because you know, for some things, it's not the whole genus changing name, it's just like 1 or 2 species. And for all those times in the past that you were like, this is Dinophysis sp, I don't know, that's gonna cause some, you know, challenges. But again, some of these are more related than others. Sometimes we can group them. Sometimes we're finding out they're very different. It's just convergent evolution. But you know, we're just trying to keep up with the literature and stuff. So that's something that's going to happen tomorrow. So just emotionally prepare yourselves tonight for those changes upcoming.
How many people like Emiliana Huxley?
I do.
Who Emiliana Huxley is, the coccolith for?
So the picture on this method.
Oh, yeah, you can take it out.
This famous sticker with the coccolithophore.
One of the most common coccolithophores. Makes these really pretty blooms. I think for people who've been doing this a long time, they're very attached to that because it's really beautiful when it blooms. It's not Emiliana anymore. And Emiliana is like a nice, you know,
Pretty,
kind of
nice.
pretty name or whatever. Now it's, yeah, very descriptive.
And I can't remember the name off the top of my head, but it's not as cute. It's not a good one.
We'll share it with tomorrow. But yeah, there's, I went to this course and I was like, this is that, and like not anymore. And yeah, even like the terms that I was using, like started process and latent process, they're like, no, we're going back to Latin, we're moving away from anglicized words. Okay, so.
Yeah. And for some things, like for a long time, we knew phallochroma, or the rotundata is an older one. So we've been sticking with Dinophysis for a long time, just because that's what, you know, we've all learned. But I think there's quite enough differences now that we're going to try and kind of shift. So first, we don't have to speak Latin in our training sessions, but it is good for you to know about it. But for the names, it's a little bit more important to stick with it. So yeah, so that'll be for tomorrow. For today, let's just enjoy these Dinophysis cultures. Oh, really? Okay.
Yeah.
Okay. There. Yeah. Oh, okay.
Did you give us 2?
Yeah. And
Yeah. And now I have to rewrite it. And then there's a split.
Do you take it from the bottom? Oh. I did, but it'll take a while for them to settle. Are there any identified here in your
yes.
You can't tell.
Yeah.
We have love.
It's full of
Food, but not Dinophysis.
Yeah. I didn't even know. We initially got ours.
Well, that's okay. Things don't always go the way we planned, and that's fine.
One out of like however many we've done, it's pretty.
I know. I'll also take this opportunity to say that this is not a comprehensive look at all, obviously all harmful species, even in our area. We just did the ones, we focused today on the ones that we could. But there's a couple that we missed, including somebody asked earlier about Protoceratium reticulatum and Phaeocystis that are 2 that we weren't able to get cultures for. And of course, there's a lot of varieties of Pseudo-nitzschia and Dinophysis that we kind of went over quickly, but don't have cultures of. We did do very thorough workshops on these in the summertime, and there are recordings of those. So if you're new or if you missed them, we can share the recordings of Anthony doing kind of slideshow intense detail on all the harmful species that you might see in this area. And we can tell you which ones are like more common and which ones are a bit less common. The target species are a good way to focus on that. Okay, Anthony thinks he might have seen, I mean, knows he saw, there's a couple of Dinophysis in here, it's just not very dense.
Maybe if you pull from the bottom, you might get more.
Should I distribute the second one?
Yeah.
The January one.
If you want to clean, and I'll give you a sample.
Okay, we're gonna distribute the second bottle that might have a little bit more in it.
Yeah, and I saw 245.
So if you have a new slide ready, Melissa will come around with those in a minute.
They'll be able to see messaging. There's a lot of that.
And if you're seeing teeny tiny things jumping around, those are the cryptophytes. And then if you're seeing medium-sized things that jump around, that will be the Azadinium. And then if you're lucky, you might see some Dinophysis. Are you seeing some, Anthony? Okay. Okay. So we'll try to share the screen for the folks online in a minute here. Down and to the left. Oh. There it is.
Yeah, there's like art.
You should look at PSI sample that you'll find.
Yeah, that's probably a good idea.
Yeah.
I sent it to Anthony the other day, so I didn't know it was, yeah, sure, I was comments, but I'm not like trying to title to just. Yes.
So clear part of
So that's a singular list from single and a solo list. Yeah. Yeah, list.
Slides. She was organized them like really well. She read it. I think she has an unknown. It's so cool to read a word like I know I'm trying to read a word. And this 60 years. It's like, oh my god, it's so cool. So I started with that. But Azadinium rubrum was in there. And she named it Space Rock. She was like, I don't know what we said, but Space Rock. So I just gave it a random name. That's like how you remember it. And she has it crossed out and it's Azadinium.
There's pictures of them coming. I mean, they all look so different, but I'm like, I don't know. Sometimes I'll get those clear ones where the cilia are coming out right out of like the end up coming right out of the cilia. The picture perfect one?
Yeah.
But otherwise,
Yeah, that's all you can do.
yeah, I'm sure there's slits and something like that.
Oh, yeah. Truly, yeah. Yeah, but
but I have some in my congregation also.
I put this one on this.
Yeah, it's not quite the next.
But I will give you a fresh. No problem. Oh, maybe every time. Take the time.
Hey folks, we've got about 45 minutes left here, and especially since this Dinophysis sample is a bit thin, now would be a nice time to start looking at the wild samples and seeing if there's any things fun in there. Several people have brought some. Maybe if you brought one, can you like raise your hand? And then that way folks that don't have one can maybe come and steal a drop. We have Poverty Bay, Bud Inlet, Camino, Quartermaster, Port Angeles. Okay, so did you guys all see that? One over here, two over here, and then one there and one back there. So if you don't have anything to look at, there's a real thick one over here. That could be fun. But yeah, so yeah, so now we can start looking at those. I think, Anthony, what are you looking at now? Bud Inlet? Yeah. Okay, and then what's on the screen now for the folks online is Bud Inlet, a sample from Bud Inlet. So we'll see if we can see anything cool. You might see Pseudo-nitzschia in some of these samples, and you might see Dinophysis in some of them as well. So the two that like looked a bit wonky in culture or didn't really show up in culture might be in here. And if you have something weird that you don't know what it is or need help identifying it, maybe you can raise your hand, ask a friend, and we can work on that together. And then also quick last call, not last call, but a quick call. If you're not attending tomorrow, come and see me for supplies. And also we have some like fun swag items. So every year Sea Grant puts out a calendar. So if you want one of those, we have some of those phytoplankton flashcards if anybody wants some of them, and we have some stickers. So if you're not coming back tomorrow, make sure you grab them. If you're coming tomorrow, we'll have them again tomorrow, and some keychains. Okay, great. And feel free to grab any food. The snacks will be back tomorrow, but the catering food, we'll have more tomorrow, but you can take it home if you want. Okay, great. Yes, they have one right there. But you guys are with me now. Okay, so they're. Oh yes, yes. We're actually wanting to
Updating our phytoplankton. I mean, oh, yes.
They all developed really aggressive. The things stress me.
Yeah.
Sorry. Never do that. They're already hard enough.
Yeah, I was talking with yeah. Yeah, I'll get her. Yeah.
Well, it's great. We both did pretty well, but I did lose one recently.
And my dad's
Our small hydrogen is on really well, big hydrogen. I was always not, yeah, we're talking.
I've never known.
Well, and I just know we sent these out for the small. Which is where I was like.
Yeah. It's like a whole fun. I can like America. Yeah.
Yell out. I'll text you.
Yeah. I just hope they're like actually. Yeah. I mean, so we make one of these things, and if we have like this, yeah. One of our,
What am I looking at up here?
They're wrong.
Yeah, no, this is Pseudo-nitzschia. And this is what we call Pseudo-nitzschia small. And I'm at 40x and zooming in visually, so I'm gonna zoom out so you can get an idea for how small it really is.
And I have some that got plastic food outside.
So this is at 20x, zoom. That's Heterosigma deadlifts next to it. Maybe look for cell size. How teeny this is. You're on 10x.
Oh, yes,
Fairly noticeable, right? That's so small. We don't really worry about them until they can be about 1 million cells per day. It's very little milk acid, but they have produced enough and density to shut down.
Right into my life. You know, it. So they actually.
This.
And then so they were able to live there, you know, and then. And they got all like the drugs, so that you could actually like try to take steps. And then there was like Angie stood up on herself. It's so exciting. And then they did that outside. And she like was like, this cow was like my life.
Yeah, he just went to
February 8th.
and the corner's like.
Yeah. And the answer is definitely.
Anybody? Not anymore. Octactus. Which it has 6 points, but sometimes it has 8, so now it's Octactus. Oh. So this is silica flagellate. So there's an animal, little flagellate, that lives inside the silica exostructure. Lots of times you'll see the empty silica structure, and sometimes you'll even see the little flagellate swimming outside or in the water. I'll go for fibula too. Speculum and fibula. I don't know
It's a struggle, because I feel the for a lot of people.
I mean, I struggled with serration way back in the day going to tripos. Some geologist found it and like, that's not fair.
Something like justify that would make sense in my head. Never seen them with 8 points.
Yeah, yeah. I know that, yeah, around here, they're always 6 points. So Octactus is just gonna be like, I don't know. Can't relate.
They can also have 4 points, right?
That's fibulate,
Yeah.
which is also Octactus.
Yeah.
Yeah, yeah. Yeah, so if the form is dense, obviously physical irritation is.
So I think so many people got fabric, so I mean, if I could get it, I'm not probably better than a dinosaur. Sorry.
Like 15 minutes over there. Or if people want to keep looking, that's great.
Hello, people online. Thank you so much for joining us today. We're kind of just like wrapping up here. But I think like no one's really manning the microscope that's sharing with you right now because we're like going around talking to people and looking at other things. So we thought this might be a nice time for you guys to take a break from your screens. And maybe we'll see you back here tomorrow for the meeting, which will be a little bit more, you know, organized and on time with like less downtime in between. And I will answer the questions that are in the chat that I just saw now before I go. And if you guys have any other questions, you can let me know. But yeah, just for everyone else that might want to leave now, we'll see you here again around 9 a.m. tomorrow. We're starting at 8:30, but we're just going to be like moving stuff around. But 9 is when we plan to start our like welcome. So we'll see you then. And so, okay, goodbye if you want to. And then for those of you that had questions, let's see, is there an equipment / supply list available? Yes, I can share that with you. Rebecca, maybe we can email about that. And Jennifer asked about the flashcards. Do you meet up with the Camino Island group? Because we can give them extra and you can have some of those there.
I do not.
You don't?
No.
Okay. Well, maybe. Yeah. Where are you located?
Whidbey Island.
Whidbey. Okay.
I could, you could give it to a Camino person and I could ask them tomorrow.
Sorry. I was trying to turn off the microphone so that wouldn't happen. Sorry, we had just had some feedback over here. Okay. Jennifer, we will give some extra ones to the Camino Island folks and maybe you can coordinate.
That would be fine.
However, we will be going to Penn Cove at some point in the near future. And so I don't know how far away you are from there. If that's easier, we could also.
Not too far.
Not too far. Okay, so that's also another option that we can be in contact about on email later if you would like.
Sounds good. Thanks.
Awesome. And yeah, thanks for joining us, everyone. And yeah, we'll see you all tomorrow. Okay, bye.